Get started. Good morning. It's good to see so many of you here this early on the first day of the conference. Really appreciate you coming. My name is Joyce Van Eck. I am the chair of the PlantGENE Steering Committee. And the way the topic for this workshop came about was feedback from our PlantGENE community. Last year, we did the basics of plant transformation vector design, and that was based on your feedback. And today, we're doing the fundamentals of modular cloning. So we're building on that information that we provided last year. So our speakers are Katie, Roger, and Josh. And then the PlantGENE Steering Committee will be participating as they can in the post-discussion after the presentations. So this is how it's going to run. We have 5 minutes for questions after each presentation. So each presenter has 40 minutes. There'll be 5 minutes for questions. We encourage you to ask questions, please. It makes it a more engaging workshop. We have a microphone, and I know it could be a little daunting, at least it is for me, to get up and walk to a microphone and ask a question. So Vina's going to run around, give her a workout this morning, ask lots of questions, and she'll bring the microphone to you. When you ask your question, please give your name and your affiliation, as I think that really helps to build community when we get to know people. And then we have PlantGENE pins. First, we were going to put them on their chairs, which I thought, well, you know, they're tables, and if you sat down and you didn't see it, not such a good thing. But we have PlantGENE pins, and we did this because we want to thank you for your support. It's a little way for us to show our appreciation, but also it helps us advertise this amazing community. We're over 1,200 members now worldwide, and we really appreciate it. Some of us have our pins from last year, and our pin from this year. So they're going to be collector's items and worth a lot of money sometime in the future. So hold on to them. So if you're not familiar with PlantGENE or the Plant Genetic Engineering Network, you can learn more about us from our website. And there's a QR code here that you can scan. But if you also go to the SIVB website, sivb.org, there's a PlantGENE button where you can get into there. You can also, please become a member if you aren't already. Membership gives you first, announcements about workshops and other events that we're having. We have a lot of resources on our website. I've provided some of them here. I highlighted the discussion forum in red. It's one of our newer resources. And again, that was based on what we heard from the community. You really wanted a discussion forum, so please use it. It's a question and answer where you can post questions, and everybody in the community gets to see the question, and it's by email prompts. Yesterday at our PlantGENE Steering Committee meeting, we decided that we're going to work on getting an app. None of us have ever made an app before, but we thought this would be a great way to have it on your phone and to be more accessible so you could get at the information a lot easier than just going to your computer. And this is my last slide. I want to thank the National Science Foundation, specifically Diane Okamura. Diane, raise your hand. Diane is a program officer with the Plant Genome Research Program. And I want to thank Diane for 2 reasons, actually 3 now I think of it. 2 reasons, because she helped myself, Piero, and Michelle, she helped guide us to get a grant funded for supporting this workshop and another one, both last year and this year. She also is a big supporter of PlantGENE, and we very much appreciate her guidance. And the 3rd reason is because we all know what's going on, the chaos, and Diane is still there. She's still pulling for us, and I want to give her a round of applause. Okay, I also want to thank the Society for giving us this platform for this workshop, and also being supportive of hosting our website, and also, again, to all the PlantGENE members for your support. This is your community, so let's make it happen. All right, thank you. All right, so our first speaker is Katie Toomey. Katie is a third-year graduate student in Wayne Parrott's lab at the University of Georgia, and her expertise is plant genome engineering. And Katie is going to give you an introduction to modular cloning. Katie? Got it.
All right. Thank y'all, everyone, for being here, and thank you, PlantGENE, for having me. Okay, thank you. So I know it's a little early. First of all, if you don't manage to take notes or take a picture of every slide, don't worry. I have a website that I have done all of that for you. Feel free to scan this QR code, and it'll take you to my little blog. Anything pertinent to this PowerPoint and everything that I couldn't fit in is on there. Also, because it's a little early, to help motivate y'all to pay attention, I made a little crochet agrobacterium. Ooh, ah. And I will throw this into the audience to whoever can answer my pop quiz 1 question at the end. So, thank y'all. All right. I don't see any more phones scanning, so I'm going to continue. I want to start with a disclaimer because I'm going to mention a lot of brands, and I have not been sponsored yet to mention them. So anything that I do mention is not necessarily an endorsement. I'm open to give my opinion on it, but I have not been sponsored in any way. So, what is modular cloning? Well, in order to understand this, you have to first understand non-modular cloning. And this is what I talked about last year, but it's essentially putting an insert into a plasmid. And I have a QR code for a little blog on that presentation as well. If you already scanned the first one, you don't have to worry about this one. So that insert is essentially, typically, a promoter, coding sequence, and terminator. Creating this non-modularly can take around 3 to 5 days. So let's say that this is your project and your PI says, I want you to change that promoter. You can't just replace the promoter if you're doing this non-modularly, so you got to reclone the whole thing, and it'll take you another 3 to 5 days. And then your wonderful PI says, I want to actually add a third promoter, and that adds another 3 to 5 days. And then when they add on nuclear localization signals or other signal peptides, well, then you've just doubled your timeline. So this would take you about 2 to 4 weeks to create these. And you'll notice that there's a lot of components that are actually the same throughout these. And you think to yourself, well, wouldn't it be just lovely if I didn't have to reclone this every single time? And that's what modular cloning helps you do. So modular cloning will help you turn those 2 to 4 weeks from just making these 3 constructs to being able to make every possible construct within that same time frame. And this is by having essentially a library of components in their own individual plasmids so that you can always find the right one at the right time. You're going to see this Disney imagery a lot. I'm a big Disney fan. So I'm getting a little bit ahead of myself, and I'm going to give you all the roadmap for this presentation. I'm going to first talk about the concepts and jargon involved in modular cloning, along with the restriction enzyme concepts you need to be familiar with. I'm then going to move into Golden Gate, what that is, what it isn't, where you can buy it, and the reaction parameters and outcomes. I'm also going to go into how to choose a Golden Gate platform, the versions available, the pros and cons, and lastly, some advice in organization that hopefully will help you not just in the computer, but also in the lab and communicate from an individual to the whole lab. So let's start with the modular cloning concepts and jargon. So a plasmid is a circularized extrachromosomal component of DNA that bacteria can take up. A vector is a plasmid that has elements allowing it to take DNA. So there's a circularized and a linearized vector here. A genetic element is what we would call just a promoter or a coding sequence or a terminator. It's just one of those elements, a component. And a cassette is all of these elements added together. It's also sometimes called an insert. A construct is what you call the vector with your insert. And the action of doing this is what's called cloning. Now, this image is actually showing non-modular cloning, but today's presentation is going to go over modular cloning. So you can see how they're different, how each element is isolated into its own plasmid. So this makes it more customizable for you, but it also means that when you're making lots of constructs, you can do it faster and cheaper. So there's a little bit of added jargon for modular cloning that you don't need to know with non-modular cloning. And that's first, a level 0 plasmid. So this is when you just have an element or a component isolated. There's also a level 1, which is when you combine your level zeros. And this is what makes your transcriptional unit. And then you have your level 2, which is where you have multiple transcriptional units. So here are those 3 constructs again that I said would take about 2 to 4 weeks with non-modular cloning. Now let's see how that would look like with modular cloning. It would take you about 3 to 4 days to make all the necessary components in your level zeros. And then once you have that library, you can just pick the ones that you need and assemble them into your level 1s. So the top is the level 0s at this point, and your bottom is the level 1s. And you can just pick different parts of your library to create all those different constructs. Now you'll notice there's a little bit of a blank here. That is what we would call a bridge, a linker, or a dummy. Each element has to have a plasmid there, but if you don't need to insert an element, then you can just use a dummy. And so on and so forth until you are satisfied with all combinations. And you could make this many plasmids in the 2 to 4 week time period. So the way that modular cloning systems work is that they'll use enzymes. Now, we talked about enzymes in my last presentation, where you have restriction enzymes that are often palindromic. These mean that they will cut at the recognition site. Here's an example with echo R1. And so because it's palindromic, this means that you can only use it once in a modular cloning system if you were to use these types of enzymes. So you would have to flank each insert with 2 different enzymes so that you can make sure that your insert is inserted in the correct direction. Now, you can use palindromic restriction enzymes to do a one-pot reaction, meaning that everything is in one pot. But with that many enzymes, it can add up. And your limiting factor for using these palindromic enzymes for modular cloning is the price point of those enzymes. So for this reason, palindromic enzymes are not really used in modular cloning. Type 2 restriction enzymes is what's far more common. And a good example of this is BSA1. It will cut actually outside of its recognition site, meaning that it leaves these nice little overhangs that are fully customizable. So they're not relevant to BSA1 being able to cut, meaning that you can have one enzyme in your one-pot reaction instead of multiple, and it will cut each recognition site, leaving different overhangs, allowing you to assemble with just one restriction enzyme, which is a lot cheaper, a lot faster. And because there are 4 nucleotides in the overhangs, this means that there's 256 possibilities of what your overhangs could be. So this allows for a lot larger reactions. However, it's also worth noting that not all 256 overhangs will react the same. I don't recommend using 4As. But it's a lot less limiting of a reaction than just using those palindromic enzymes from earlier. And here's a QR code to an Agene blog about enzymes if anyone's curious to know more. Like I said, the most common type 2 restriction enzyme is BSA1. So it's relatively cheap at about $80 a reaction. It can be heat inactivated easily. And when you're using it for modular cloning, it takes about, on average, 30 seconds a cycle, so it's relatively quick. Systems will typically use a secondary restriction enzyme for their reactions as well. So the three most common are ESP3I, AAR1, and BPI1. So these are a little bit more expensive, and they take a little bit longer than BSA1, which is why BSA1 is typically more commonly used. But you need a second enzyme if you're hoping to cycle, as we'll get into later. So now we're going to talk a little bit more about the Golden Gate. So if you've ever heard of Golden Gate or gateway reactions, they kind of sound similar. So I want to preemptively just say that they're not the same thing. The Golden Gate is dependent on those type 2 restriction enzymes that I mentioned earlier, and gateway is actually dependent on recombinases. So Dr. Thilmany will go into recombinases a little bit more in his presentation, but just know that they're not the same thing. And gateway is a very wonderful cloning technique that I will not be going into. So when you're creating your level 0, you can use non-modular cloning techniques to create this level 0. But when you're creating the level 1s and 2s, you have to use the modular cloning techniques that I'm going to teach. So that's what this Golden Gate reaction is about. You'll use the BSA1 to have your multiple different level 0s digested, leaving those overhangs that we talked about earlier. And I'm going to throw in a dummy sequence here, just so that y'all remember that it exists. And then in your one-pot reaction, they'll be able to ligate to each other based on those overhangs that are unique. And they'll also be able to ligate, importantly, to the backbone. And again, I mentioned that there are 256 possible overhangs, so there's a very high ceiling for how many inserts you can have. NEB on their website, the largest reaction that I could find was actually a 52 insert assembly. So if you're worried about those 9 or 8 inserts, don't. So there are 2 kits that I was able to find through NEB for cloning purposes. You can use NEB Bridge, which includes the BSA1 enzyme. So if you're only using BSA1, this would be a good option. But it is about $9 a reaction. So I'd recommend the bottom kit, which has just the ligase master mix. It doesn't include your enzyme, but you can buy the enzyme separately. It's relatively cheap. And so this will equal about $3 a reaction, so a lot cheaper. So as I've mentioned, you'll have those level 0s, your elements, your components, plasmids, and they'll typically have a selection such as AMP or chloramphenicol. You'll also have an acceptor. In this case, I'm using a kanamycin selection, and you'll notice that it has a CCDB gene. So this is a toxic gene that will help select against an undigested backbone or acceptor. And the CCDB gene is what will be replaced by your inserts. So once you add your appropriate DNA to your reaction, all that's left is adding a ligase and a buffer and a restriction enzyme. And that's all. So then your Golden Gate reaction will undergo cycles of being digested and ligated roughly 30 times. And then you just transform into E. coli. I recommend NEB either 5-alpha if your construct is less than 10 kilobases, or NEB 10-beta if it's more than 10 kilobases. And after you transformed into competent E. coli cells, you can plate your reaction. And let's take a look at what the possible outcomes would be. So we're hoping that we get the correct assembly and that they get picked up by E. coli to replicate. However, life doesn't always work that way. So it's important to remember that you just put DNA into those tubes, those plasmids, and not everything is going to get digested, and not everything is going to get digested perfectly. So on your plate, you may have lots of plasmids or components that did not get picked up by E. coli. And when you're doing your screening, it's important to keep this in mind so that you don't pick up a plasmid that has only one of these elements and think that you have a positive reaction when you don't. So sometimes you'll find just a partially digested element or a partial assembly without a backbone, or a linearized backbone with some of the inserts. Now, none of these have an intact ORI, so they won't be replicated by E. coli, but if you're unlucky when you're screening, you might think that you have a positive colony when you don't. So let's do away with those for now and look at some of the other options. You could have an undigested acceptor, but like I said, it has that CCDB gene, so this shouldn't be too much of a concern. You'll also potentially have some undigested level zeros, but because they have a different selection than the plate that you're using, because remember, we were transforming into kanamycin selection, should minimize the concern for that, so that hopefully the only thing that is assembled and picked up by E. coli be your perfectly assembled construct. Now, it's hard to tell which of these E. colis will actually have it. So just remember that when you're doing your PCR screening, that you shouldn't use primers that are just in one of the inserts. You can use ones that would have that would span multiple inserts, but what's most recommended is using one primer in the backbone and one primer in your insert. So, the part that we've all been waiting for. Let's go into the Golden Gate platforms, the versions available, the pros and cons of them, and how to choose. So, we're not quite done with the jargon. I do have to talk a little bit about multiplexing. So multiplexing is what I've been talking about earlier when I said that there's a one-pot reaction. It just means that you're able to do multiple reactions in your one 2-milliliter tube. So there's your organization of assembly, which you can either do in series or in parallel. They both start with your library and end with your final construct, but how you get there is a little different. So if you're cloning in series, you have to finish cloning plasmid A before you can clone plasmid B, before you can clone plasmid C. When you're cloning in parallel, you can clone A, B, and C at once and use this to get to your final plasmid. When comparing Golden Gate platforms, it's also important to decide if you care about having cycling. This means that if you have your final plasmid and you determine that you don't actually want it to be your final plasmid, are you able to add more elements, or did you clone yourself into a dead end? If you're able to add more elements, and this means that the system is compatible with cycling, and that you can keep recloning and cloning till your heart's content. So, the 5 versions that I'm going to be going over in terms of Golden Gate are Golden Braid, Mobius, Moclo, Green Gate, and Multigreen. Dr. Thilmany will also be going over gantry system, which is a type of modular cloning. However, it, as I mentioned, is recombinase-based. It's also agro-based. So all the ones that I'm going to go over are type 2 restriction enzyme-based, and they're based on cloning in E. coli. So you'll have to still transform into agro. So, Golden Braid. Golden Braid allows for 3 level zeros, 3 components, into your level 1s. It does use 2 enzymes, BSA1 and ESP31, so that you can clone into either your alpha or omega levels. Now, I know I mentioned levels 1 and 2 earlier, and this can be a bit confusing. So they actually use their own terminology that they don't really call level 1s and 2s, and we'll get into why in a second. They do allow for multiplexing with this platform. As long as you're using 2 transcriptional units or fewer, you can clone in parallel. But if you're using more than 2, so if you have your Cas9 construct, your guide construct, and your selection, then you'll have to do cloning in series. So my personal notes on Golden Braid is, in my opinion, I think it's a bit unintuitive. The unique terminology makes it a bit, it adds to some of the confusion. And you also still will need to transfer to a binary when you're done cloning so that you can actually move into agrobacterium. So the way that cloning with Golden Braid would work is you have your 3 level 0 components that you digest with BSA1 to create your level alpha, which you'll notice that there's an antibiotics selection swap here so that you can select for your level alpha. You can use 3 new level 0s to make a second alpha, and then you can use ESP31, to digest these 2 alphas to create an omega. Now, the reason they don't use level 1 and 2 terminology is because they can also allow for you to use these level 0s to clone into an omega. So it's not really a level 2. They both allow for you to clone into either of these starting points, alpha or omega. And then it does allow for cycling because you can use that BSA1 again to cycle back into an alpha acceptor. These presentations are about to get very colorful. I'm sorry, I kind of run out of colors at some point when I'm making like 10 plus assemblies. So now let's move on to Mobius assembly. If you didn't like that Golden Braid had only 3 components per level 1, then you're going to like Mobius because it has 10. I'm not going to go over how they define each component because it's right here, but 10 components is a lot more flexible if that's what you're looking for. The enzymes that they use are BSA1 and AAR1. They do revert to the typical syntax of having a level 0, 1, and 2 plasmid. They opt for using AAR1 because it has a longer recognition site, so they say that it makes it less likely that you'll see that recognition site in one of your constructs or in one of your inserts, so less of a concern in that matter. It does allow for multiplexing. You can clone in parallel as long as you have 4 or fewer transcriptional units. So if you're doing that situation I described earlier with your Cas9, guide, and selection, you could use Mobius to clone in parallel for that. I have a lot of notes for Mobius. I think that with all the components, it does have a bit more of a learning curve. However, there is also some pretty colors to Mobius. They don't use CCDB, and instead they actually use visual confirmation with proteins like GFP, so you can see if your construct transformed or not based on if it fluoresces. With Mobius, unlike Golden Braid, you can't go from your level 0 to your level 2. And you still need to transfer to a binary after you're done cloning in Mobius. So I didn't want to use 10 level 0s. I ran out of colors at some point, so here's a 5-component assembly that they use chloramphenicol for their level 0 selection. And then you can transform into a level 1 with while selecting for kanamycin. And once you have 2 level 1s, you can clone them together with AAR1 to create your level 2. And you can cycle with Mobius. Moving on into MoClo, it's a lot more beginner-friendly of a modular cloning platform. It allows for 5 components, your promoter, your 5' untranslated region, signal peptide, coding sequence, and terminator. The enzymes that they use are BSA1 and BPI1. They do have 3 levels, so this means that they are allowed to multiplex, and they actually have the greatest number of acceptors for your level 1s or 2s, so you can clone up to 7 transcriptional units in parallel without having to worry about cloning in series. It's worth noting that BSA1 and BPI1 cut differently, so it's just something to be aware of when you're cloning in silico. And you'll also, again, still need to transfer to a binary before you move to agro. So y'all have hopefully gotten a little used to looking at this diagram by now. You understand that we're looking at a level 0 that you clone with the first enzyme to get to your level 1. And you can do this again so that you have another level 1, and you can clone them together with your BPI1. And you'll notice that there is another antibiotic switch here, so you can clone from level 0 to 2, and you can cycle. Now, GreenGate is the first of the platforms I'm going to talk about that you don't actually have to transfer to a binary, and we'll discuss why. It allows for 6 components, so it's a good middle ground between the 10 and the 3 that I mentioned earlier. You have your promoter, a signal peptide on either your N or C terminus, a coding sequence, a terminator, and a selection module, so you can have your plant selection as a part of your assembly. Unfortunately, it doesn't allow for multiplexing because they only use one enzyme, so they only have level 0s and 1s, and there's no multiplexing at all. At least no straightforward multiplexing. It's a good balance in terms of the number of components that it can take, and it has the borders for aggro, so it's ready to go into aggro once you clone. And its biggest downfall, like I said though, is no multiplexing. So your level 1s will have the borders for aggro and the ORI compatible for aggro, but once you get to that point, it's a dead end. So to overcome this main hurdle of GreenGate, someone in my lab recently came out with the multi-green expansion kit of GreenGate. So it still allows for the 6 components because it's using the same syntax as GreenGate. However, he added an additional enzyme, ESP31, so that you can clone into level 2. And there's a good amount of acceptors, so you can clone up to 6 transcriptional units in parallel without worrying about cloning in series. Didn't mean to capitalize those ED there, but I must admit that I am biased because this was made by my lab mate. My personal opinion is that this is the best. It does allow for the borders of aggro, it allows for multi-level cloning, et cetera, et cetera. So, as I said, here's your level 0s. You clone into your level 1s, and once you have multiple of those, you can use ESP31 to clone into your level 2, which already has your borders for aggro and the ORI so that you can transform into aggro directly. But the special note is that your level 1s will also have those same borders and ORI. So if you, for some reason, decide that you want to use just one of those level 1s for a control, you can do that. So I'm going to show the most complex plasmid that I've personally used multi-green to clone. So here's a construct that I've used for prime editing. It's 23,000 base pairs. And one of my level 1s has, if there's ever a blank, that means that there's a dummy there. So one of my level 1s is 8,000 kilobases for my prime editor. Another level 1 has my guide. My third level 1 has a mismatch inhibition protein. And then I can use a level 0 linker/dummy for this cloning, like I said. So you can do multi-level cloning, meaning you can use your level 0s and 1s to get a 2. My fourth level 1 is visual selection, and my 5th is plant selection, so that I can create this massive construct relatively easy. There was a lot of trial and error for me with this one, but it did take me about 2 weeks to get it once I had my level 1s in place. And it has the ORIs the ORI and borders ready to go into aggro. So I just threw a lot of information at y'all. So I made a table so it'd be a little bit easier as a take-home. In terms of modular assembly sizes, the important take-home is that Mobius has the most and Golden Braid has the fewest. In terms of number of levels, all these cloning platforms have 3 except for GreenGate. The number of acceptors, MoClo has the greatest. If it's intuitive, based on my opinion, I would say that MoClo, GreenGate, and multi-green are the most easiest to understand. And only GreenGate and multi-green have those borders and ORI for aggro. And For my opinion of what's the best and worst traits of each of these, I'm not going to read all these out. You're welcome to take a picture of this. It's also on the blog. But I'm going to once again say that in my opinion, multigreen is the best. I couldn't really think of a worse trait for it except for, I guess, perhaps that it doesn't have the visual selection, which is not necessarily needed by any means. It's just really pretty to look at. So I've mentioned that visual selection a few times. This is what it looks like in Mobius. So you can see when there's a positive event, it'll actually change color. And it's a rather stark change. It's really pretty to look at. So I also have a flow chart to determine which Golden Gate platform is best for y'all's lab. The first question you have to ask yourself is, do I need modular cloning? If you're only ever working with one construct really and you're just changing guides, it's not necessary for you, but I applaud you if you want to get into it. The next question I would ask is, how many elements, how many level zeros do you need for your level 1 assembly? If you need 3 or fewer, Golden Braid will work for you. If you need 4 to 6, then Green Gate, Multigreen, and Moclo will work. And if you need more than 6, I feel so sorry for you, but you'll need Mobius. In terms of how many transcriptional units do you need for your final plasmid, this is synonymous with how many acceptors each platform has. So Moclo, as I said, has the most. And if you want your final construct in cloning to be aggro-ready, as I've said plenty of times, Green Gate and Multigreen are the only ones that have compatibility with that. So I was asked to give some words of advice on setting up with modular cloning in your lab. And what I would say is once you choose a platform, stick with it, because most of them are not compatible. The syntax of the overhangs and the antibiotics just don't really make, they don't play well together. So only Green Gate and Multigreen are actually interchangeable because they do have the same antibiotics and overhangs. But aside from those, I wouldn't really try to combine others. Any of the vectors that I've mentioned today or any of the platforms that I've mentioned today do have availability through AdGene. And then, of course, you would need to order all necessary reagents like the kits that I've said, and the enzymes, and the antibiotics. And don't forget, if you're using CCDB, that toxic trait, you need to use chloramphenicol with it. So my last module in this talk is advice and organization. And I'm going to talk about how you can help clean up your computer and lab. So this is what my cloning folder looks like. Essentially, I have a folder for tools and then a folder for each project. Under tools, I will have everything under the sun that I will use for cloning. So I'll have my acceptors / backbones, my level 0 library, my level 1s and 2s that are ready for plant transformation, any dummies / bridges / linkers, any primers that I use as well, and I have a folder that I keep just for checking, meaning it's something that I'm currently digesting or PCRing, and then my raw sequences that I get back from any sequencing platforms. And then, of course, under each project, I'll have the notes for it, the level 0s and the level 1s, and the final plasmid. So this is personal to each person, but what's really important to know that everything that I make, I upload to the cloud. Or another platform where everyone in your lab would access it would be just as well. But the important thing is that we want to make sure that everyone in our lab would be able to access anything that I make. So everything that I upload to the cloud includes plasmid maps, so they know, like if someone wants to use AmCyan as a selection, they would know that I've already made a plasmid for that they don't have to remake that. Plasmids can be stored in what's called a freeze stock. It's a glycerol stock, and we have the strains that are in our freezers in a Excel online, and we also note their location on our OneDrive as well. And we'll also keep our primer sequences and locations also noted online so that no one has to remake a primer. So by the end of this, you'll have not just your level 0 plasmid library, but also your free stock library and your primer library as well, that everyone will be able to access and hopefully makes cloning a little bit easier in your lab. And so for that, I want to say thank you to my PI and my cloning mentors. Thank you for attending, and stay in touch. I have my LinkedIn and the last chance for the blog. All right. Are y'all ready for the quiz?
Oh, first quiz. Let's do the quiz.
Sorry. What is the most common type 2 restriction enzyme? I'm fine if you shout it out or if you want to run to the mic.
Oh, okay.
BSA1?
Yeah? Okay, hold on.
Throw it.
Ready?
If you drop it,
Thanks, Katie. We have time for questions for Katie. And also a reminder, when you ask a question, please give your name and affiliation. And we've timed this so that at the end, we're going to have roughly 45 minutes for questions. We're going to bring the speakers back up, and you'll have an opportunity to ask questions, but also share your experiences. It makes it, a lot more engaging. And remember, Vina's going to run around with the microphone, and it looks like Daniel's got a question.
Hi, this is Daniel, assistant professor at the University of Maryland. So I think one thing that you might have overseen on the presentation, which might seem obvious, is that when you use Golden Gate cloning, actually matters that whatever you level 0 you put in, that part doesn't have to have those same restriction sites, because you have to domesticate it, right? So that's one of the disadvantages of all of the Moklo systems. The other one, and I'm not trying to make any commercials to Moklo, but you might have missed some key points of that. The first one is that it's not just 7 parts. You can actually have level P's, level M's, level 2, 1A, and so on and so forth. So we actually made a position 1, level 1 with 5 different genes by using these subsequent 0 cloning. So in my experience, Moklo is actually better than all of the systems we have tried in the past, just for the reason that you can be so flexible than just, you can reclone previous parts into the initial parts and make more complex constructs. So far, our largest construct is close to 30 kilobases, and it's just 1 position. So we still have 6 more.
thank you. Any other questions? While I walk there, if you are in social media, LinkedIn or something, please post about this session. How did you like it or something like that?
Hi, Greg G. from Oregon State. So related to that point on the domestication side, I just thought I'd ask about your thoughts about doing site-directed mutagenesis versus ordering synthetic fragments, and whether you take a hybrid approach or just everything is synthesized in your lab, and trying to find a good balance point myself a lot of the time.
Thank you for that question. So the question was, is it better to have a hybrid approach or synthesize everything or try to clone everything? So it's kind of dependent, obviously, on what you're working with. If I'm trying to domesticate something and there's 3 domestications within like a 40 base pair range, I wouldn't really want to PCR that, so I would probably synthesize the fragment depending on the size of it. But, I mean, if it's just 1 domestication site, that's probably something you could solve with PCR. I think it's a case-by-case basis.
Questions? Okay. I'm going to get a lot of steps. I need it.
Hi, thank you very much for the presentation. It was great. I really appreciated the approach to it. I wanted, first was just a comment to add in, and the second was a question. The first We had also noticed that there's like a GreenGate version 2 now that had incorporated some of the items you had said it was missing. And it was also our understanding that it was still compatible with the multi-green and the old GreenGate kit, if you had any comments on that. And then the other, you had mentioned like the size of the plasmid that you're working with yourself. I was just wondering what levels of efficiency you were getting in terms of using it downstream.
So to address the first comment and also the first comment that was given after this presentation, I am aware that there are multiple expansions to these. Golden Braid has overcome many of the disadvantages that I've listed. However, I could not go over absolutely everything. So there's more listed on my little blog, but I just went over the first introduction of each of these platforms. So if you are interested in one but you didn't like just a particular disadvantage that I listed, it may be worth to still look into it and see if more publications have come out on it. And to address your second point, I have only cloned it. I am still actually working on the plant transformation side.
Hello, my name is Hannah Levengood. I am a fourth year PhD candidate at Purdue University, and I have one question that's directly related to this and another one that's a little bit more fun. But the first one is, when you actually are considering which inserts and stuff to use for each one, do you also need to consider like the size of the inserts? Because I know that there's some genes that are very large, like the Ruby gene, that I've heard can be difficult to clone. And my second one is, would you be willing to share your pattern with how you made the crochet agrobacterium?
Yeah, I can put that on the website. I just have to write it down first because I actually made that on the car right here. It's a long drive from Athens. But to address your first question, I'm sorry, I actually got really caught up with the crochet. Can you repeat the question?
Yes. So I'm just curious to see how this large size of the inserts, would impact
sorry. So there is actually a formula I use for how much of each insert I will use in a Golden Gate reaction. I didn't put that on my blog, but that's a good thing that I should. Many publications will actually say how many nanograms they recommend per size of construct. So check with each publication, but I can certainly put the one that I use on the website.
Good morning, and thank you everyone for attending this workshop. It's my honor to introduce our next speaker, Dr. Roger Tilmony from USDA Agriculture Research Service. So Dr. Tilmony has extensive expertise in gene expression, molecular biology, tissue culture, and crop bioengineering. So today he's going to tell us about efficient gene stacking using gantry system.
Thanks, Kinsub. Thanks everyone for coming this first session of the meeting, and I'm very glad to be here. It almost didn't happen. So today I'm going to talk about a system we developed in my laboratory called Gantry. And it's a modular cloning system, and it's going to add to some of the great introduction that Katie gave on Golden Gate cloning. So first I'll tell you what's Gantry. So it's a technology that you can use to simultaneously introduce multiple traits into transgenic plants. And the postdoc in my lab, Ray Collier, came up with the name Gantry, and it stands for, it's a modified acronym that stands for Gene Assembly in Agrobacterium by Nucleic Acid Transfer using Recombinase Technology. I can't even remember that. I have to look at it to make sure I say it correctly. But it's actually an analogy, which is pretty memorable. And actually, this is appropriate here in Norfolk because it's also a port. So what you see in this picture in the red and white is a gantry crane. And so the gantry system is an analogy to that gantry crane that can load large amounts of cargo onto a container ship. And so the analogy is the ship is agrobacterium, and the gantry crane is the system for loading it. And so our motivation for developing gantry, which actually started many years ago now, was we were thinking forward and wanted to have the capability of modifying complex multi-gene traits or engineering novel metabolic or signaling pathways in transgenic plants. And to do that, we expected that we're going to require the introduction of multiple genes, not just 1 or 2 or 3, which is typical for constructs that people use, but rather stacking multi-gene traits together in the plant requires specific mechanisms in doing that. And so you could do it in a bunch of different ways. You could build simple constructs and use multiple rounds of transformation to introduce them one at a time into the transgenic plant sequentially. Or you could breed multiple transgenic plants together to bring the traits all together into one plant. But we thought the more efficient way to do it is to use an assembly process and transfer a single stack construct into the plant all at once. At the time we started this, the Golden Gate cloning methodologies were just coming online, so it wasn't very obvious at that point that there were easy ways of making these large stacked assemblies. And particularly at that time, stacking together more than 3, 4, or 5 together expression cassettes was quite challenging. So we set out to develop a system that allows simple and reliable transgene stacking and the efficient production of transgenic plants. So gantry is quite a bit different than the systems that Katie talked about, and I'm going to point out some of the unique attributes of this system. So transgene stacking is not performed actually in a test tube. It's performed in vivo in the agrobacterium strain that you're going to use for transformation of plants. This method enables the sequential stacking of multiple transgene cassettes into a single tDNA. And that tDNA is actually located in the native virulence plasmid of that agrobacterium strain. So it's not on a binary vector, it's not on a binary back vector, it's in the genome of the agrobacterium on its virulence plasmid. Actually, it replaces the native tDNA with your synthetic or introduced tDNA. And this technology, as Katie alluded to, uses some novel, very powerful enzymes called unidirectional site-specific recombinases. And the 3 that are used in the gantry system I list there, they're PAR-A, TP901, and A108 from the phages that they came from in bacteria. And PAR-A is a unidirectional exciser. It recognizes these 2 identical sites called MRS, or multi-resolution sites. And when they're in direct orientation, what the PAR-A recombinase does is it'll excise that intervening sequence, leaving behind just a single MRS site. TP901 and A108 recombinases, they're large serine resolvases. They perform unidirectional integration between an at-P and an at-B site. They recognize these 2 sites and then perform integration, integrating that circular donor molecule into the recipient blue linear strand shown there. For both TP91 and A118, the recognition sites at P and B for each of these enzymes are completely different. So TP91 recognizes the red sites and A118 the yellow. And so this is a sophisticated system, but it utilizes just 3 types of components. It's actually quite simple for the user to use gantry for stacking. The first component that you need to make the gantry system stacking work is a modified agrobacterium strain. The original strain that was developed we call R-Port1 because it's a derivative of a rhizogenese strain. And what you can see on the left part of the slide there is it's been modified, disarming that agrobacterium strain, so the native tDNA has been removed and replaced by gantry components. And I don't know if it's large enough for you to see there, but the components include an introduced left border region, then an at site for A108 recognition, and then a kanamycin selection marker. So the original strain doesn't have a complete tDNA, it just has a kanamycin selection marker. The other 2 types of components you need for gantry assembly are, first, you need 2 donor plasmids. And I don't expect you to see all the details of these two maps, but all you need to know about these plasmids is that they're typical cloning vectors. They have a large multiple cloning site. The original versions of these donors have a large multiple cloning site for traditional restriction enzyme cloning. But I'll tell you more about other donors that we have that you can use now instead of these. All the user has to do is clone their sequences of interest into either the B or the P donors so that they can be stacked using gantry. The last type of component that's needed for the system are two helper plasmids. We call them helper because in this case, the user doesn't have to modify these plasmids. These two plasmids simply provide the recombinase enzymes for gantry stacking. And they're in another cloning vector that can replicate in E. coli, but they don't contain an origin of replication, so they cannot replicate in Agrobacterium. And I'll tell you more why that's important in a little bit. So you just need these three components: the strain, donor vectors with your cargo sequences, and then the two types of helpers. So the original donors we created were just for traditional enzyme cloning, restriction enzyme cloning, but since we developed the system, we realized everybody has switched to using Golden Gate cloning or gateway cloning and other things. So we also modified the donors to be recipients for those types of cargo. So this shows three different plasmids we have that are gateway cloning recipients. The one on the left is just a generic recipient that can take any cargo that has the right at sites. The other two are expression cassettes where you could insert your coding sequences of interest either behind the 35S promoter if you want to express it in dicots, or in the maize ubiquitin promoter if you want strong constitutive expression in monocots. And more recently, we've developed Golden Gate cloning recipient donors. We adopted the MoClo system in my lab, so those are the ones we have now, but it would be easy to modify the BNP donors to be a recipient for your favorite Golden Gate assembly cloning library that you have. So what's shown here are the two level 2 recipients. And the reason I'm showing you level 2 is that it'll allow you to stack multiple transcriptional units or expression cassettes into these level 2 donors. So you don't have to stack an individual expression cassette one at a time using gantry. You could actually stack 6 or 7 expression cassettes into a single B donor or P donor and use that to introduce that into the gantry strain. And if you want to stack more than 6 together, you could put 6 in the B donor and then another 6 in the P donor, and you could actually make a 12 stack with just 2 gantry assembly steps. We also have level 1 MoClo recipient donors, so if you just wanted to do one expression cassette at a time, you could do that as well. Okay, so I'm going to walk you through sort of the details of how the process works. Don't worry if this doesn't make a whole lot of sense. You can always figure out these details, or you can talk to me later about it. But this is the basic method by which the gantry assembly works. So you start with our original modified strain, R port. You then take that strain, make it into competent cells, and co-transform it with 2 plasmids, a B donor that contains some cargo of interest for you, and a B helper. So the original strain is kanamycin resistance. The B donor is bringing in gentamicin resistance. So when you co-transform these 2 plasmids into the original strain, what happens is the helper expresses the 2 needed recombinases, which perform integration of the plasmid, and then an excision event removing the prior selection marker that was in the RPort1 strain, making a new strain now that has your cargo sequences of interest within a tDNA, and the strain is now gentamicin resistant, and it lost its resistance to kanamycin. So you could actually use this stack 1 strain directly for plant transformation. But you can also take this stack 1 strain, which is now gent resistant, and it has an at site, the red small arrow shown there near the right border, and do another round of gantry assembly with a P donor. So the P donor plus the P helper, so with your new cargo in that P donor plasmid, you'll do another round of assembly where you get integration and excision to create stack 2. And now it'll have both cargo 1 and cargo 2 sequences within the tDNA. And of course, if you're paying attention, we've now toggled back to kanamycin. And we've left behind an at site for another round of B donor entry into the system. So you can iteratively repeat this process as many times as you want to build a tDNA theoretically as large as you want with as much cargo as you want. And so that's the basic process of how it works, but this is what the user actually is doing in this. So you're just making competent agrobacterium cells, transforming it with two plasmids, a B donor and a B helper, and then plating those cells on gentamicin. You're then picking colonies, restreaking them on sucrose. Why sucrose? Sucrose, we use it as a negative marker to confirm excision of the previous kanamycin selection marker. And then you can pick those colonies off the sucrose plate, and just to make sure that you know what happened, restreak it on kanamycin and show that it doesn't grow, but it does grow on gentamicin. You can then take those cells, make them competent, and do another round of gantry assembly with a P donor and a P helper. The same process, you're selecting on kanamycin first, restreaking on sucrose, confirming that it integrated and excised the cargo, and then repeating that process over and over again. So if you can do agrobacterium electroporation transformation, you can do gantry assembly. So if you're interested in the details of this process, we published a book chapter in Methods in Molecular Biology, and you can look up for all the working details of how to do this assembly. So when we initially made this system, we wanted to test to see how well it works, how well does this work for building large stacks and for transforming plants. And so Ray initially built what we call a 10 stack using 10 rounds of sequential assembly. And that built a tDNA that was over 28,000 base pairs in size with 8 expression transgene cargos. And they're all listed here. I'm not going to walk you through all of these, but I'm going to point out a few details. So it shows the expression cassette that was in each of the B or P donors there on the left, the phenotype that it conferred in plants. The reason we chose these cargos is because they all had easily detectable phenotypes in plants. So it was very easy to phenotype the plants we're generating using this construct. And then it shows the total size on the right. There are a couple important details I want to point out. You'll notice the promoters, coding sequences, and terminators are listed in the expression cassette on the left. We didn't repeat the same promoter more than once. We did use the same terminator several times just because we didn't have a lot of terminators at that time to choose from. But the reason we chose different promoters is important. If you want to stack multiple genes together into an expression cassette, I'd strongly discourage reusing the 35S promoter 3 times or 4 times. That's not a good idea. For one thing, it'll make a repetitive sequence in your assembly that could affect the stability of the construct, as well as induce silencing, and then you get silencing of all those traits if it's transgene silencing based on that promoter sequence. So using a different promoter and different control sequences, terminators, to express your genes of interest is an important attribute to think about. The other one is that the first cargo that we inserted was actually a selection marker. It was stack 1. It was the 35S promoter on this SUL1 gene, which confers resistance to an antibiotic, sulfadiazanine. The reason we put a selection marker near the left border is because that's the last part of the tDNA that gets transferred from agrobacterium. So if you use selection for that last cargo sequence, you're ensuring that presumably a complete tDNA got transferred from the agrobacterium into the plant. You'll notice we also use a selection marker near the right border, which is the last cargo 10 there, which is an NPT2 or a kanamycin resistant marker. And I'll show you how we use both of those together. And so this is just a diagrammatic representation of what this 10 stack looked like. But of course, the question is, how does it work when you transform plants? And so, sorry, I'm getting ahead of myself. When we initially developed this system, what we wanted to do is we wanted to make sure it was doing the assembly exactly as we expected. And so at that time, the easiest way to validate that the strain had the cargo that we introduced was to use genomic PCR on the agrobacterium strain itself. And so what we did is we designed primers that both annealed within the cargo sequence and in neighboring sequence within the strain. And that's what's shown by the rectangles with the blue numbers below the construct assembly there. This is just the primers that we use to interrogate for the presence of all the cargo sequences we introduced. And so this is what the gel images looks like for all those amplicons, confirming that the 10 stack strain that we created had all these cargo sequences. And so we did this actually not just at the last step of the 10 stack, but we did it at step 1, 2, 3, all the way along. And following each stacking event, the new agrobacterium strain was validated, and all the colonies that we checked had correct assemblies. So the fidelity of the stacking system is high. We also, an interesting attribute of the gantry system, it's not based on a binary vector, which is an introduced plasmid into the strain. It's actually part of the genome. It's part of the native virulence plasmid, which makes these constructs in agrobacterium extremely stable. We actually grew the 10-stack strain in rich media, LB, without antibiotic selection, kanamycin. And then after 3 days of growth, we plated it out for single colonies just on non-selective LB and checked 100 of those colonies on kanamycin plates. And all 100 of them were still kanamycin resistant. Then Ray actually went through, I don't remember how many he checked, more than 20. He did the PCR for all 10 of the cargo sequences, and he validated that all 10 of those colonies were kanamycin resistant, and they still carried the entire 10-stack construct. So the stability of this construct in the strain is very high. I guarantee if you build something large like this into a binary vector or a buyback vector and then grow it without selection, 3 days later, a lot of those colonies will not have the plasmid anymore, right? Okay, so, all right, now I'm getting to the point that I jumped ahead for, which is how does it work in plants? Well, the easiest plant to test when we initially were testing this system is, of course, Arabidopsis with floral dip transformation. So what we did is traditional floral dip transformation using the 10-stack strain. And then we took the T1 seed and plated it on 2 selection agents simultaneously, both kanamycin and sulfadiazonine. And of the seed we plated, 1% of them were resistant to both antibiotic. And this is just, Ray went through and tested all the phenotypes in these plants. And it just shows you examples of the phenotypes that we see in the plants. Of course, we selected for sulfadiazonine resistance there on the left. We detected firefly luciferase activity, green fluorescence in the pollen, herbicide tolerance by bar, gust staining, MIB anthocyanin-induced expression in flower petals and seeds, red fluorescence in the whole plant with TD tomato, and then kanamycin selection. And if you collated all that data from over 400 independent events, 90% of those lines were positive for all 8 phenotypes. And you can see the actual percentages there shown in the table. We also looked for if any of those lines, carried sequence outside the tDNA. So if you do binary vector transformation, what you'll routinely see in transgenic plants is sometimes you'll get part of the plasmid vector integrated into your transgenic plant. We looked at a number of these events, and 93% carried only the tDNA and no sequences outside the tDNA. The next thing we went to characterize these plants is to look at the copy number of the introduced construct. And to do that, we used a droplet digital PCR method to examine the copy number of 3 components in the 10-stack. The first component we looked at was the sulfadiazonine selection marker. And of the plants that we tested, over 68% of them had just a single copy of that part of the 10-stack. It was a little bit different story if we looked at the copy number for the GUS gene, which is located in the center of the 10-stack, and the NBT2 marker, which is near the right border. And what you can see, there's a clear trend there. The copy number for the NBT2 sequence was definitely higher, but still over 27% of them had a single copy of NBT2. But there were also 2 copies and more than 2 copies as shown in the graph there. Then we went on and to characterize not only these events that we selected using both kanamycin and sulfadiazonine selection, we took some of the seed and plated it just on the single antibiotic selection markers. So we selected a population that was just growing on kanamycin alone, just sulfadiazonine, or the dual, which I already talked about. And what you can see in the 2 colored lines, green and blue, so the dual, I already showed you all that data. Interestingly, even if we just select using only sulfadiazonine, which is the marker near the left border, what you can see is that over 90% of the plants still had all 8 phenotypes. And that's because I'm getting back again to putting your selection marker near the left border. It ensures that a complete tDNA transfer had to occur to deliver that part of the construct. And so that worked really well. What you can see in stark contrast to that is if you used a marker near the right border alone, in this case kanamycin, the trend is very different, right? And it's pretty linear, actually, surprisingly linear, how it goes down from you selected for kanamycin, so that's obviously at 100%, but by the time you get to the sulfadiazanine marker near the left border, only 65% of those plants had also sulfadiazanine resistance. And so I'll talk a little more about that in a minute. So Arabidopsis is a great plant model, but what my lab primarily works on is in crop plants. So we wanted to know, how does gantry work in crops? And so we collaborated with, I had colleagues there that showed how gantry worked in potato, but my lab had long-standing interest in working in making transgenic rice, and so I'm going to present the data that we generated in rice. So we couldn't use the 10 stack that we used in Arabidopsis in rice, in part because most of the promoters we used on the genes of interest in the 10 stack were primarily functional for dicots. They wouldn't function that well in a monocot grass like rice. And so we built a new assembly this time, and this time it's actually considerably bigger. It's over 37,000 base pairs in length with 11 cargo sequences. But again, we picked expression cassettes that give us easily assayable phenotypes in the transgenic plants. And so that's what's shown here. And then we generated transgenic Nippambari rice plants using the gantry 11 stack strain. And this just shows you some of the phenotypes that we observed. We selected using hygromycin, which is the selection marker near the left border of the 11 stack assembly. But we also looked at Ranilla and Firefly luciferase activity, Gus staining, which is expressed only in green tissues, herbicide tolerance, both glyphosate and glufosinate resistance, as well as green fluorescent in roots, primarily the root tips. And that just shows you examples there. And then we characterized these 37 events for the quality of events that we recovered. And what's shown here is the diagram of the 11 stacks on top. Below that, you see these rectangles again. These are, again, representing PCR amplicons that we used, in this case, genomic DNA from the rice plants to interrogate for the presence of specific sequences in the transgenics. And what's below that in color is, if you see the color there, that means it was found present in that particular event. And so none of these, the first thing I'll tell you is none of these 37 events had any backbone sequence outside of the tDNA borders. So again, it's only delivering the tDNA predominantly into these plants. Half of the events, 51%, had all 8 of the assayable phenotypes. So, and based on genomic PCR, had a complete 11 stack tDNA. So half of the 37. 22% of these were what I refer to as high quality events, because they had not only a complete tDNA, but the tDNA appeared to be single copy because we looked at the copy numbers of the HPT2 selection marker, the Gus Plus gene, and the promoter on NPT2, which was maize ubiquitin. And so I'm going to focus on, actually, since we published this work, I'm going to focus on, we went and characterized 3 of these events more thoroughly. Event 9, 42, and number 4. So 9 and 42 are what I call high quality events. They were apparently single complete tDNAs. And number 4 was clearly not that. It had cargo sequence from near the right border side of the tDNA and cargo sequence near the left border, but it apparently was missing sequence in between those. And so we used long read PacBio sequencing, first to look at 11 stack number 9. And I'm just showing diagrammatically of what we found from that long read sequencing. We validated that it contained a complete 37 kilobase 11 stack tDNA. It was only missing 281 base pairs near the left border. And obviously that wasn't a functional loss because the HPT2 selection marker still worked in this event. The tDNA was inserted into an intragenic region of rice chromosome 6, and it caused a very small 64 base pair deletion. So this validated for this event, it's a high quality event in that it contained the entire construct, expressed all the desirable phenotypes, it didn't inactivate a native gene in the genome, and it had the complete tDNA. It was a similar story for event number 42. Again, it had almost the incomplete tDNA. It was missing 285 base pairs near the left border. And It was also inserted in an intergenic region, this time in chromosome 9, with a very small deletion from the genome 17 base pairs. So both of these events have been validated with long-read sequencing to be high-quality events based on this data. So the story for the more complicated result of not containing a complete tDNA is event number 4, and that one has 2 partial tDNA insertions, both apparently in the same chromosome. A 17.2 kb and a 19.9 kb portion of the tDNA was separately inserted into chromosome 2. It's about separated by 9.5 million base pairs. We couldn't actually identify the integration sites on one end or the other of those partial tDNAs, but we definitely located in chromosome 2. The second partial tDNA was inserted into a gene, so it actually inactivated a gene in that particular event. So I was gratified to see that what we had discovered before, that this was an event that didn't contain a complete 11-stack tDNA, was validated with the long-read sequencing. But again, we validated 2 of the high-quality events were complete tDNAs as well. And so some questions I sometimes get is, so what is the actual capacity of the gantry system? How big can you build these tDNAs? So you've already seen the data from Arabidopsis. It's just been regraphed here on the upper left part. I can now add the data from the rice plants. I showed you data from the 11-stack, but we also built a 5-stack and tested it, and they're just graphed onto this graph. The 5-stack is a little outside of where we'd expect based on the Arabidopsis data, but if you draw a line, a regression line through the Arabidopsis data, it runs right through the 11-stack data, and if you extrapolate it out, it looks like the carrying capacity, or not necessarily the carrying capacity, but the delivery capacity of our strain would basically stop working above about 76 kilobases in size. But that's not actually the capacity of what you can stack. We've now built, I'm not going to go through it, but we've built very large tDNAs, including ones that are bigger than 76 kb. Gantry can build it, but the agrobacterium strain is not efficient at delivering extremely large tDNAs into plants. So I'm just going to summarize the rice data. About half of the events that we detected were high-quality events that had a complete tDNA. 42% were the complete tDNAs, and none of the events had vector backbone. So I've shown you Arabidopsis, rice. I mentioned we also had done potato, but the point here is that those aren't the only species gantry can be used in. So my lab has tested it in Brachypodium switchgrass. My colleagues have tested it in citrus, tomato, and we've recently also used it in grapevine. I know other groups have used it in soybean, wheat, corn, poplar, pennycress, and I'm pretty sure there are probably other species out there. My advice in regards to this, if agrobacterium transferation works in your species, the gantry system will likely work in your species as well. It might not be the same efficiencies you're used to, because it might be dependent on an optimized agrobacterium strain for your particular protocol, but we not only have the original rport1 gantry recipient strain, a colleague of mine, James Thompson, has developed an EHA 105 derivative, as well as other agrobacterium strain derivatives that are also gantry recipients. And so you could always make your own strain if you wanted to. And of course, the reason I'm partly here is to tell you that the gantry system was created with public funding. It's available for distribution. We distribute it with a simple material transfer agreement. All you got to do is email me and ask me for the MTA. And the thing I'm telling you right here is that the MTA only restricts you from distributing the original materials that we share with you. If you build donor vectors with your cargo sequences, you put them into a gantry strain, that strain is not ours, that's yours. You can do with it what you want. And then if you create transgenic plants from it, the USDA, I don't own it, you own that. So the only thing that's limited by the MTA is the original materials. And the reason we asked for that in part is because I just like knowing who's using our system to do whatever they're doing, and that's one way I know who's receiving the materials. All right, so just to summarize, I hope I showed that gantry can be an effective and easy to use technology for stacking genes with an agrobacterium tDNA. It enables the assembly of large stacked arrays. I showed you lots of data for a 37 kilobase 11 stack, but it can even go larger than that. It frequently generates transgenic plants that contain a complete single copy tDNA integration event, and they could be used in a wide array of plant species. And of course, I didn't do all this work myself, so the three highlighted in blue are members of my own group. Ray Collier is a former postdoc in the lab. He now works at the Wisconsin Crop Improvement Center. And Layla, she works at a company, but she developed Ray did all the work in Arabidopsis. Layla did all the work I showed you in rice. And Tony's done the work in multiple other species that I presented today. And this was also a collaboration. James Thompson contributed a lot to the original development of the gantry system. And then, of course, we worked with our other colleague, Kent McHugh, to show its utility in potato. And it was, of course, supported by Agricultural Research Service funding. And if you're interested in learning more about the system, you're welcome to come and talk to me, but we've published most of this work, and the publications are listed there. If you search using gantry, I'll tell you you'll find it, because it's a pretty unique name. But with that, I would be happy to answer any questions.
Questions? Yeah. Can you introduce yourself?
Hi, great talk. Annie Saltaricos, I'm from Plastomics. A quick question, I understand why you were using the antibiotic selection to look at basically trying to gauge if you have full insertion of your tDNA. Have you ever tried not selecting using an antibiotic, no selection whatsoever, or using one of your visual markers, and did that change, or could that change some of your results?
Yeah, we've not tried to do that. You could certainly use a visual marker in your selection. The most recent work we've done using gantry and grapevine, selection is more like a suggestion, I would say, than a selection. So we use a red fluorescent reporter in that case, and then we're physically picking explant material to take forward because of its red fluorescence. I haven't done a comparison to see if that affects the transformation efficiency calculations, but I could see it definitely could on how effective that particular marker you're using is, yeah. Yeah, the rice transformation, I can't imagine doing that without selection. I think that would be hard. Arabidopsis, you could probably do it, but you'd have a screening event to do it without selection, yeah.
Question here.
Yeah.
Hi, Roger. Yeah, Beth Rowan, Oregon State University.
Hey, Beth.
My question is more of a technical one about the PacBio sequencing. So I was wondering if you predetermined the location of the insertion sites and then you just threw the whole thing in there and then you did simultaneously discovery of the insertion site and the characterization. Okay.
Yes. No, it was de novo. We just did the assembly and then determined the site based on that alignment of the assembly of the PacBio reads. And they were really long, so that was pretty easy to do, especially for the high quality events. For the more complicated partial events, it's a little harder, especially as you noticed we didn't actually locate the other integration site for that particular partial event that I showed you. But for high quality single copy complete tDNA events, that works great. Yeah, it's very simple to figure out what happens just by looking at the sequence read data, basically. So
cool, thanks.
I want to ask you a question. This is Veena from Danforth Center. Can you give people here a feel for how much it would cost them to PacBio sequence one event?
I don't know if I have the perfect numbers. We did it about a year and a half ago. It might be even less expensive now, but I think it was between $800 and $1,000 maybe to do a couple events. So they were pooled together and then deconvoluted separately from that run. So yeah, and it also depends. If your species has a small genome, you can pool many more events together and get enough sequence data to deconvolute it all. If the genome, say if it's an hexaploid wheat, that's a lot harder to do because the genome is so much larger, yeah.
One question here.
Hi, I'm Leonard from VIB in Belgium. I was wondering, could you hypothesize why you almost never see backbone integration in your system?
Yeah, it would be a hypothesis. I don't definitively know an answer. I think it comes down to a couple reasons, likely. One is it's the tDNA is in the native location of the native virulence plasmid that's part of the genome of the agrobacterium strain. So it's the delivery vehicle for tDNAs. It's evolved to be that, and that might give us higher fidelity in the way the processing of the tDNA occurs in the agrobacterium and then its subsequent delivery. The other thing is that most binary vectors are multi-copy in agrobacterium, and so they tend to deliver a lot more tDNA. That's my impression. And the more you deliver, the more likely it might fail to terminate near the left border and then deliver some vector backbone. And then there's a third hypothesis. I've never tried it, but if we built a really small tDNA with, say, just a single selection marker in gantry, I think we might detect more transfer of DNA beyond the left border, because I think the native system evolved to deliver a normal-sized tDNA, which is, if you look in the native agrobacterium strain, tDNA sizes run around 20 to 30 kilobases in some strains. And so if you build a really small one, I don't think the system is ready to stop yet when it gets down the road 5 kb from the right border initiation. And so I certainly think the bigger tDNAs, you're going to get a lot less transfer beyond the left border, just because the system is ready to stop when you get to the left border, when it gets to that point. Those are all hypotheses, but I think they're reasonable. I don't know how testable they are, but they're reasonable explanations. Yeah. Thanks.
While Amy set up here for the next speaker, please note that this recording of this will be available and accessible through PlantGENE after some period of time, so it will be there, all of these.
Thanks, Vina. Yeah, just to follow up, so we will have the recording, but it won't be available until September. So it'll be a little bit of a wait, but we'll get that to you guys as soon as we get it from SIVB. So I'm Amy Malzahn. I'm excited to introduce our next speaker to you, Josh Clem. Josh Clem is a PhD candidate in Dr. Yiping Chi's lab at the University of Maryland, College Park. Since studying molecular biology as an undergraduate at Frostburg State University, Josh has been fascinated with plant biotechnology and its utility in improving crop productivity into the future. Josh decided to join Yiping's lab at UMD to focus on plant genome editing technology development, where he leads several projects on novel CRISPR-Cas systems and reproductive trait engineering.
Okay, good morning, everyone, and thank you, PlantGENE in particular, and Joyce and Amy for the invitation to discuss some of the research that I work on in Yiping Chi's lab at the University of Maryland, and also thank you, Katie and Roger, for excellent introductions to the cloning strategies that I'll be going into a bit more, and also for Roger for introducing the gantry system. So in the spirit of modular cloning, I'll be discussing our system for assembling tDNA expression constructs for using CRISPR in plants. So just to introduce CRISPR, we're all quite familiar with CRISPR. Obviously, it's a genome editing tool capable of transcriptional activation and as well as several other strategies. Just to give an outline to the presentation, I'll just introduce CRISPR. I'll discuss kind of our principles for assembling tDNA expression constructs. I'll provide a workflow. I'll discuss various components of our toolbox, how we organize our systems, our vector toolkits, and I'll also discuss a bit our AdGene resources that are available to the community. So just to provide a very brief introduction on CRISPR, and certainly not a comprehensive introduction. CRISPR is often used for knocking out genes in plants, either through canonical or alternative NHEJ. We can also perform gene knock-in, where you insert DNA sequence into the plant genome. We can perform base editing to install single nucleotide substitutions in plant genomes. We can also perform prime editing for kind of allelic replacement, and this has also been recently adapted for introducing inversions and deletions in plants as well. We can perform transcriptional regulation to control gene expression. We can modify the epigenome of plants using CRISPR as well, and we can also do RNA editing. So the bolded letters in our lab are what we particularly work on, what are very well established in our lab. So knocking genes out with CRISPR tools, base editing tools, as well as tools for transcriptional regulation. And so we have an entire genome editing workflow involving rice protoplast transformation, as well as agrobacterium-mediated transformation in several plant species in our lab. However, I'm just going to focus sort of on what's circled in red here, just the first component, which would be vector construction. So just some basic considerations for constructing tDNA vectors for expression in plants. First, you might consider what nuclease you want to use. And when considering nucleases, there's various Cas nucleases. You want to think about the editing efficiency in the system that you'll be working with, the type of DNA repair outcome that you're interested in. So whether you're interested in doing substitutions, or if you're interested in just knocking genes out, the nuclease that you use matters for these strategies. Also, the intellectual property landscape around these systems are different, right? Because Cas9 is a bit more complex than Cas12a, for example. Moreover, you'd want to consider your guide RNA expression strategy. So whether you're interested in expressing your guide RNAs under sort of a constitutive polymerase II promoter or a smaller polymerase III promoter. And finally, you would be interested in considering what promoter you'll drive your CRISPR machinery with, your Cas nuclease or your enzyme, based on the species compatibility and the expression profile of that promoter. And we have modular assembly toolkits available for each of these kind of components, which I'll get into further. So I'm going to discuss our cloning strategy, which is actually kind of a hybrid system between Golden Gate and gateway assembly. And we first published this back in 2015, so it's very well established in our lab. And everything that I discuss beyond this will kind of be further adaptations to this system. So we've just been sort of building on this system, which has been very popular in the plant genome editing community. And we've tested this system, our lab and other labs, in a variety of species, both monocots and dicots, including rice and Arabidopsis and maize, wheat, tomato, citrus, and even carrot, and we're expanding this all the time. So primarily, I work on rice in the lab. And just to get into a broad overview of our strategy, it involves kind of three main steps. So the first step would be, after you design your guide RNAs, you would insert those into what we call guide RNA entry vectors. And we don't use the typical kind of level 0, level 1 grammar for our system. We just refer to them, the vectors, more intuitively as they are guide RNA entry clones. So you anneal those oligos first, and you would insert those into PYPQ 131 all the way up to 136, for example, depending on how many guide RNAs you're interested in using. So in this example, I have PYPQ 131 through 134. So that would be used for 4 guide RNAs. And that would be inserted into, just based on restriction enzyme digestion and ligation of those annealed oligos, into a backbone of PYPQ 144. So 144 for 4 guide RNAs. If we were using 2 guide RNAs, for example, it would be 142. And then you can screen that using actually blue-white screening. And then the final step after that's a Golden Gate reaction to combine all of those guide RNAs, is going to be combining that with your nuclease clone, as I'll break down these steps further in the following slides, along with a destination vector, which we combine the 3 of them with gateway assembly to assemble your final tDNA expression construct. So the first step being annealed oligo cloning. So this is an example if you have just a single guide RNA. You would combine these oligos together in a pot. You can phosphorylate them, and you can there are a few ways you can do it. You can boil them and bring them down to room temperature to reduce their secondary structure and to anneal them, or you can just use a thermal cycler, for example, to bring it from high temperature down to low temperature. You anneal them, and then you would digest this backbone, PYPQ 131, for example, using BSMB1, which is an isochisomer of ESP31. And actually, ESP31 tends to work better than BSMB1 in my experience. We get that from Thermo Fisher Scientific. Works a bit better than BSMB1 from NEB. And once you insert your annealed oligos containing your guide RNA into this guide RNA entry clone, these have tetracycline resistance, and then we would simply screen them using Sanger sequencing, the commonly used primer. And this is a very efficient reaction. Typically, you would only need to screen just one colony. This annealed oligo cloning is very efficient. So the next step is Golden Gate. So if you think back to the annealed oligo cloning step that I just showed you, you would do that for each of the different guide RNAs that you have. So if you're using 4 guide RNAs, for example, you perform that annealed oligo ligation into 4 different guide RNA entry clones. And in this example, it's PYPQ 131 through 134 for 4 guides. And then you combine that into a single tube for Golden Gate assembly into the PYPQ 144 backbone using the most commonly used restriction enzyme for Golden Gate assembly, BSA1. And this allows for blue-white screening, so it's removing the beta-galactosidase gene. And typically, the colonies should appear white, and this is another very efficient reaction. You would only need to screen 1 colony in most cases. Up to many guide RNAs, just a single colony almost always gives you a perfect clone. And then finally, so we've gone through the steps of creating our guide RNA entry clones, right? So with 4 guide RNAs. So that would be this vector at the top, PYPQ 144, harboring guide RNAs 1 through 4. And then we have 2 other vectors that we need to combine into constructing this final tDNA expression clone. The first would be the nuclease vector. So in this example, I'm just using Cas9, and we all love Cas9. It's the most efficient Cas nuclease. So this vector is called PYPQ 167, and I'll show the add gene deposited vectors here in a moment. And then a destination vector would hold your plant selection marker, and it also contains the CCDB toxin, which is then removed during the gateway recombination reaction. So you just combine these 3 vectors, and you typically incubate it overnight, about 16 hours or so. And this, again, is another very efficient reaction. So the final tDNA construct harboring the promoter driving Cas9, as well as the several guide RNAs, in this case driven by polymerase III promoters, U6. We have different options for that, polymerase II or polymerase III promoters, as well as your plant selection marker. And these typically run around 20 kilobases or so. You would almost always just need to screen a single colony for these. And what we often do, now that we have whole plasmid sequencing available to us through companies like Plasmid Saurus, is we just send the whole plasmid. Previously, you would do a digest and maybe use multiple Sanger sequencing reactions. And because the system I described is most efficient with up to 6 guide RNAs, for example, we've also expanded this for up to 16 in what we call higher order assembly, and I won't go through all the details of this, but essentially it's a combination of the Golden Gate reaction and just basic restriction enzyme digestion and ligation, and we go through several steps. And we've actually tested this in rice, and we've achieved highly efficient genome editing with up to 16 guide RNAs using Cas12a. And so I just want to provide an overview of how we assemble these constructs in silico, because it's very important to first do everything in silico before you do it at the bench, right? And we use SnapGene. There are many options for this. There's Genius. Also, Benchling is capable of many of these steps. But SnapGene is just one that we use and we really like. So the first step is going back to the annealed oligo cloning, and you would simply, after synthesizing these as oligos, harboring at either side the restriction enzyme overhang, you would anneal them, and then you can, anneal them in silico, and then we'll move on to the next steps of the in silico step. So once you've annealed them, you can then use restriction enzyme digestion and ligation of your guide RNA entry clone to ligate them. And this is, again, easy to do on SnapGene based on your annealed oligos that you've done. And then finally, you can perform the Golden Gate assembly also in silico on SnapGene. So SnapGene, again, isn't the only way to assemble these constructs in silico. Most others also, there's some free softwares as well, like Plasmid Editor, that can likely are capable of this. And then the final step is the multi-site gateway, which we use LR clonase 2, combining the guide RNA entry clones with the nuclease vector, in this example, harboring Cas9, and the gateway destination clone, which is PYBQ 203. And that's it. So that's how you simply engineer or construct these vectors in silico using SnapGene. Very straightforward. So next, I just want to, since I've given an overview of our cloning strategy, I want to get into some of the common nucleases or common components of our toolkit. So the most basic is Cas9, and we all know this is the most efficient Cas nuclease for use in plants. Part of Jennifer Dowden's lab is actually interested in understanding why it is that this particular SP Cas9 is just so efficient compared to other Cas nucleases. But this is the most commonly used, and we have a few options for this. The first one would just, the most commonly used one in our lab at least, is PYPQ 166, which is a maize codon optimized variant of Cas9. And we also have Cas9P for plant codon optimized, and we typically use this for expression in dicots. So Cas9, of course, often results in insertions and deletions, usually single nucleotide or single base pair insertions or small deletions very efficiently in plants, and it uses this 3' protospacer adjacent motif, or PAM, of NGG. Secondly, probably the second most commonly used one is Cas12a. So we have two options for this. We have just wild type Cas12a, which is pretty efficient. However, we also have an engineered variant Cas12a harboring 3 mutations, RRV, which makes it more temperature tolerant and also seems to improve the overall genome editing efficiency. And there are some differences in Cas9 and Cas12a, of course. Usually with Cas12a, you get slightly larger deletion outcomes. Also, the PAM site is actually at the 5' position of the target site for Cas12a. And rather than NGG, it's TTTV, so more of an AT-rich PAM. And sometimes this is ideal for targeting AT-rich PAM sites, such as promoter regions, for example. Next, we have Cas12b, just a slight variant of Cas12a with a slightly different PAM preference, TTN. And we've also harnessed this one for transcriptional activation, unlike Cas12a. So this is just another separate option as sort of an alternative to Cas12a. And then we also have what we call PAM unconstrained nucleases built off of Cas9. So these enzymes have been engineered in labs to essentially interact with non-NGG PAMs. So SPRY, for example, will recognize PAMs of NNN, so any nucleotide, right? And NG similarly recognizes NGN. And this is a result of mutations in the PAM interacting domain of Cas9. There are about 11 mutations here. And these tend to be actually quite efficient in plants, at least in rice. However, there are some problems with off-targeting given that it sort of promiscuously binds DNA and just kind of is always searching for PAMs, but it tends to recognize everything. So it takes a longer time for it to actually find its ideal target site, and that's likely what results in sort of the lower editing efficiency compared to just wild-type Cas9. Another issue with these is that they actually can self-target, for example, because the tracer RNA, the first 3 nucleotides, are GTT, and those happen to be recognized by this nuclease. So it can self-target, and that's something that we're interested in addressing to sort of improve this, to reduce this activity. And we also have C2T base editing tools for installing substitutions. In this example, I'm showing you Cas9 is fused to a cytosine deaminase, which will deaminate the cytosine, convert it to uracil, and then through mismatch repair, you end up with a C2T transition mutation. And Cas9 is also fused to a uracil glycosylase inhibitor, or UGI, to prevent excision of the A-basic site during deamination. And this particular deaminase is one of our most efficient deaminases, this A3A, Y130F. This is also available on AdGene. If you just go to AdGene and you type in PYPQ265, for example, this will pop up, and you can view all of the plasmids in our lab. And this has a deamination window, which is shown here. Typically, the first 5 to 10 nucleotides are deaminated in, using this deaminase. We also have A to G base editing tools, and this one is fused with a TAD-A8E adenosine deaminase. This one is also quite efficient and can confer A to G base mutations, and these are often used for engineering herbicide resistance in plants. We also have CRISPR combo. So this enables simultaneous genome editing and genome activation. And this is based off of the idea that when you truncate the length of a CRISPR spacer, so the guide RNA, when you truncate the length of that, it actually abolishes the nuclease activity of Cas9. So we can use that to still search for a target site and bind DNA, but it will not cleave the DNA. And so that's how we can actually combine that with genome editing in this, what we call the CRISPR combo system. We can combine this with genome activation, which is based on the recruitment of many activators fused by SunTag to a certain target site. And in this example, we targeted baby boom 1, the morphogenic regulator, and through genome activation, we're able to produce shoots in a hormone-free way. And we can couple that with base editing or NHEJ outcomes. And we also have what we call an all-in-one tDNA expression method. So this is brand new. This is, the purpose of this is actually that it's not really modular. The idea is that all you have to do is insert your guide RNA of interest into the backbone of the plasmid. So if you can compare it to our conventional gateway system, there are several steps in that, from annealing the oligos, inserting them into guide RNA entry clones, and then going through Golden Gate, and then also going through Gateway. The idea of this is just to make it much more simple for the community, and that you start with a backbone that's pre-assembled. It already has the CRISPR machinery in it. It has your promoter and selection marker of interest, and all you have to do is insert your guide RNA. And this is work by Yanhao Cheng, a former postdoc in our lab. And this is just a system that's made to be more simple, and we can also screen guide RNA libraries by sort of combining them in a pool and just ligating them into the backbone and then introducing those through agrobacterium into plants. So this is great for library screening as well as for simplistic tDNA vector assembly. And this is just an overview of the steps that it would require to insert your guide RNA into here. So you start with your pre-assembled backbone harboring all of your CRISPR machinery, and all the user is responsible for is designing their guide RNA and ligating that into the pre-assembled backbone. And we've designed this system to be expressed in very well in both monocot and dicot systems. And this is just an overview of the capabilities of the all-in-one system. So we have Cas9-based mutagenesis. We also have cytosine base editors available, adenine base editors available for this system. CRISPR-Act 3.0, which is just our CRISPR activation system. And we also have adapted this system for use with Cas12a and even a Cas12a base editor. And so just to give an overview of our AdGene resources, I went through not an entirely comprehensive list of our plasmids. We have over 300 deposited on AdGene, but if you just go to AdGene and type in Yiping Qi, my PI's name, you'll find all of the vectors that we've deposited and the associated publication that comes along with those vectors. And so this is my -80 glycerol stock. So it's not actually my glycerol stock, just a representation. I promise this is actually how I organize it. So I personally like to keep separate boxes for nuclease vectors. So there's Cas9, Cas12a, those PYPQ 166, 230. I like to keep them in separate boxes from my guide RNA cloning vectors, which would be all of the 130 level vectors, the Golden Gate entry clones, and the destination vectors or recipient vectors. And finally, I like to keep a box for the gateway destination vectors harboring the plant selection marker and the promoters for driving the CRISPR machinery, all in separate boxes. And then we have a shared lab box, which would contain just the common constructs that we use in our lab. And these are freeze stocks or glycerol stocks, keep in the -80. We also keep all of the in silico assembled constructs and copies of all of the plasmids on AdGene through this box folder, which is a secure storage or cloud method that UMD provides to us. And we keep all of the commonly used constructs, for example, in this folder titled Common Constructs in the Qi Lab. And I also want to shout out to our website. So if you're free to visit this, it just contains all of our AdGene resources, some information about what our lab does, and some also news about our lab when we release publications, and some photos of us as well. And with that, I think I've concluded all of our modular system for CRISPR assembly. So I just want to thank my PI, Yiping Qi, as well as my lab members in the Qi lab. I've linked to our website, the AdGene materials, as well as my LinkedIn here. I've also provided my email. Feel free to reach out to me, ask any questions, and I thank you for your time.
Questions? Oh, there.
The back there.
I'm going to check QP update.
Thanks. I'm Ken from Oregon State. So you mentioned 16 guides in one construct. So two-part question. One is, what's the most guides you've transformed in a single experiment? And of the guides that you know actually work, that are actually like good guides, have you noticed that introducing more guides at the same time reduces editing efficiency?
Yeah. So to my knowledge, that's actually the most that at least the most that we published in our lab. It was 16 guides in rice. And to answer your question, we achieved, and that was with Cas12a, highly efficient genome editing, so high frequency of biallelic mutations at numerous target sites, using 16 guides. And to answer your second question about the efficiency, it is well known that when you increase the number of guide RNAs that you're using, it tends to slightly reduce the editing efficiency at the other target sites, likely due to just a dilution of your reagents, essentially. However, with the most efficient editors like Cas9 and Cas12a, that's not a major problem. It's just when you get into the less efficient systems like base editing, prime editing, that it becomes a bit more of a problem because they're already limited in the genome editing efficiency as a whole. Yeah.
Hi there, I'm Freddie from Purdue University. This was a great talk, thank you. My question is very similar to the one that was just asked about guide RNAs again. We found success in our lab having multiple guide RNAs for our same target gene. Have you been seeing any of that in your experience with this work?
I think that's a very, I guess I'll describe it as a safe way. So if, for example, if you want to knock out a gene, it's not always guaranteed that single guide RNA that you design is going to work, right? So it's a good idea to include maybe 2 guide RNAs targeting the same position. Well, not exactly the same position, but the same gene or allele, for example. And there are a few ways in which you can assess the activity of a guide RNA prior to deploying it in plants. One would be sort of an in vitro method, where you look at the cleavage activity just using an RNP based on a plasmid. That doesn't translate extremely well to systems because it doesn't factor in things like chromatin accessibility, for example. So the other way to assess the activity of a guide RNA would be like a protoplast strategy, right, which we use in our lab, and that's one of the reasons that we use it, is that it enables us to actually test the functionality of different systems prior to going through the months that are required for stable transformation. But if you are interested in knocking out, say, a single gene or even multiple genes, it's a great idea to use more than one guide RNA. And when you do that, sometimes you get dropout events, we call them, in which everything intervening the 2 guide RNAs is removed, and that can lead to a very robust knockout phenotype. So that's a good strategy, yeah.
Hi, this is Ritesh from Boyce Thompson Institute. Actually, my question is regarding miniature Cas proteins. Have you any experience with them, and how's the efficiency in terms of?
Yeah, other labs have, like TNPB, I think it's, for example, is a much miniaturized ancestor of Cas9. Other labs have had good success with these. We have had some success with using a much smaller mini Cas12 variant called Cas12F or Cas12J2. It's far less efficient than the predominant Cas9, Cas12A systems. I personally actually didn't have success using Cas12J2, but I know other folks have, or for different target sites it has worked. So if it's if you're interested in using viral vectors, for example, that would be much more amenable than like Cas9 or Cas12a, just based on the smaller length of the sequence. But I don't have too much experience using them. Yeah.
Question here.
Yeah.
Hi Josh, nice presentation. Just a quick question. Since your lab has multiple Cas proteins, I wonder if for all the testing you're using the same elements, the same promoters and terminators, and what's your take on actually using different promoters to drive the expression? Do you think that would somehow boost efficiency on those that are not that much, or do you think it's more of a protein issue?
Yeah, so when we're comparing nucleases, we always use the same promoters, of course. So when we're preparing or comparing different strategies, we would, of course, use the same regulatory elements to drive them, so it's not biased. So there are certainly differences in use of promoters, and monocots and dicots in particular. We've had a lot of success with maize ubiquitin promoter and monocots, and that's the one that we typically stick to. And we also use that same promoter and monocots to drive the guide RNAs, right, under a POL2 constitutive promoter. In that case, we would use a method to separate the guide RNAs after they're transcribed, such as the tRNA method or the hammerhead HDV ribozyme strategy. However, in some other species, dicots for Arabidopsis, for example, I probably wouldn't use one of those constitutive promoters. I might opt for something like an egg cell-specific promoter to increase the frequency of heritable mutations. Other than that, monocots, maize ubiquitin works really well for us across different species. Yeah.
Hi, Josh. Thank you so much for your talk. My name's Claire. I'm a graduate student at UC Riverside. So I had a question about LB-Cas12a. What temperature do you find during the transformation makes the editing most efficient, and at what time point in the transformation do you apply that heat? Because I know typically the LB-Cas12a, you need heat stress.
Yeah, so using a heat treatment for your genome editing experiments is a pretty surefire way to increase the editing efficiency. I don't personally use any heat treatments because we're interested in comparing nucleases just kind of at a baseline, so we don't always do a heat treatment. For Cas12a, it is a lot more temperature sensitive, so that's why it's kind of challenging to use it as a nuclease in systems like Arabidopsis. But in rice, for example, we do our selection at 32 degrees Celsius, and that seems to be perfectly amenable for achieving efficient genome editing outcomes using Cas12a. And I think most of the editing, at least my hypothesis is most of it happens during rice selection, because that's when we slightly elevate our temperature. And then we go back down to 28. Yeah. I think I answered your question. Okay, good.
Thank you. My name is Pankaj Bhomik. I represent the National Research Council in Canada. This was very interesting, to learn all the recent updates. So I have a quick question for all three presenters. That considering the customization, improving the efficiency, multiple guide RNAs, different way of clonings, different plant species, different enzymes. So do you all think that the recent example of OpenCRISPR, whether the large language model or anything that generative AI could be deployed, we have generated a lot of data in terms of learning about cloning, efficiency. Just an open question, the thoughts that are you thinking about that, or are you aware of anyone interested in discussing this?
OpenCRISPR, yes. Actually, Joyce, I think you posted to PlantGENE the blog recently asking if anyone has used OpenCRISPR. So to my knowledge, no one has actually used OpenCRISPR for genome editing in plants yet. And I am sure there are people that are actively working on it and are interested in doing it. But I don't really have any further comments on that because, to my knowledge, it just hasn't been tested. We haven't personally tested it either. Yeah.
At least in public domain.
Exactly.
Then that could be one suggestion for PlantGENE to consider, that who are interested. I'll be very much interested because from the National Research Council, we are working on that. We received a funding in terms of deploying artificial intelligence because of recent government's priority area. So that's something I'll request that PlantGENE, if you would like to consider to discuss more.
On Joyce's behalf, sure.
I'm going to help you. I'm going to grab the other microphone. So let's thank Josh for his presentation. Okay, so now we're going to switch to, we have time for open discussion, and we're going to ask our speakers to come up on the stage here. And you have time to, you can go back. Just stay up there, Josh, to answer your questions. And I have some information to help guide you through how we're going to run this. Thank you, and I'm going to help you. Okay. So we had 3 fantastic presentations, with Katie talking about an introduction to modular cloning, Roger giving the gantry system overview and detailed information, and then Josh on CRISPR. So what we want to do now is we have time for open discussion. So these, it can be questions for our panel, anything that they've spoken about, if you need more guidance, more clarification. But what we like to do for these workshops is to have you also share your experiences, what worked, what didn't work, your frustrations, because that really makes it a lot more engaging and helps people to learn. We know that some people join late. If you'd like to move up, you're welcome to. Vina and I will both run around with the microphones. And we know that sometimes people can be a little bit it's difficult to get up maybe not the courage, but I know when I was a graduate student or student, it was hard for me to ask a question. So what we did was we set up a Mentimeter option that you can scan the QR code, or you can use that URL, type in your question, and Amy's going to read them and ask the questions for you. This is something that somebody shared with us. They did it at a conference, and they thought it was really successful. So we're going to do that, too. We'll give it a try. And Amy, our very efficient Amy, emailed the PlantGENE community with the link, so you can even simply go to your email. So you have 3 different options. You have your email, you have your QR code, and then you have the URL that you can type in. Now, again, the recording of this workshop, as are all of our workshops, including the one we held last year here, are available on PlantGENE. But this particular video won't be available on PlantGENE till September. It will be available on the SIVB site for those of you who register to attend the meeting. But there's kind of this period where it's only on the SIVB site, but it will then be on the PlantGENE site. And Amy will send an email to everyone when it's on the PlantGENE site, but again, you can access it through SIVB. All right, are we ready to get started? Okay, so again, you can ask him a question in person, give your name and your affiliation, or also type in your question. All right. Vina.
I would like to ask speakers the question, if I was a beginner and then I am trying to do, thinking about cloning or CRISPR or complex modular gantry system, can you can you give a piece of advice that you wish you knew at that time when you were thinking about doing it, it would have made life better for you? So it can be very silly, very basic, so that nobody feel people can start asking basic questions. So there is no dumb question here. So you have it.
So I think we all probably have a piece of advice that we would want to give. One thing that I would say that I didn't go over in my presentation is, like Josh said, it's very important to do in silico cloning before you do your cloning at your bench, because I know one time I got ahead of myself and I cloned something, sent it off for sequencing, and I had the in silico after I did the cloning at the bench. And I found that apparently when I combined the entry vectors that I did, it'll form an artificial start site. So you need to be aware of definitely doing your in silico cloning and checking that out before you clone at the bench.
Yeah, I would second that from Katie. Doing the in silico first, so you know for sure that you're going to build what you actually want and need, and it doesn't have any unexpected outcomes. The other thing I would say, if you're really new to this, there are a lot of systems out there. It's worth doing the research in the front end. I mean, between what you were exposed to today, there are certain systems that are better adapted to being used for particular functions, and finding the one that works best for what your needs are is really important. I was a slow adopter to Golden Gate cloning, and there were a lot of systems by the time I finally decided to go down that path, and they're not all interchangeable, and that's really, after you make that commitment to go down a particular path, it's really hard to change after you've done that. So making that choice very conscious in the way you proceed, I think, is a bit of good advice.
Yeah, so I agree with all of that, obviously, and understanding how to perform in silico assembly is absolutely crucial to understanding your system, for one, and also just ensuring that you're using all of the correct components and everything is as it should be. another thing which maybe we didn't bring up as much is just you shouldn't always trust the integrity of your plasmid. So if, for example, you're struggling with assembling a construct, it might not be your fault. It might not be something innately wrong with what you're doing. Nowadays we have resources like Plasmidsaurus, which uses nanopore-based DNA sequencing that you can use to just sequence your entire plasmid, right? Which is much better than just using Sanger sequencing. And another thing, if you're short on funds, for example, because it's about $15 to sequence an entire plasmid, you can perform a restriction enzyme diagnostic digest to just as a preliminary check of the integrity of your plasmid. And then finally, going on to the agrobacterium stage of things, I'm not sure if a lot of other labs do this, but what I typically do to check the quality of my binary vector once it's in agro is I actually mini prep it out of the agro, which doesn't work very well, but it yields few enough copies of the plasmid that you can then transform that into an E. coli strain such as DH5 alpha, and then mini prep it out of DH5 alpha and send that for whole plasmid sequencing. So then you have sort of an indirect way of assuring the quality of your binary vector that's already been transformed into agrobacterium. And I find maybe about 10% of the time in DH5 alpha that I get actually plasmid rearrangements, which you really want to avoid, obviously. So that would be my advice. Yeah.
Thank you.
We have a question back here.
Hi, Josh. Nice presentation. I have used a couple of like Epping's plasmid, labs plasmid. Most of the case 12A plasmids, they don't have like any fluorescent marker. When you do and go for a transmission, it's difficult. And also monitoring is very easy if you put a like fluorescent marker. Second question is, what is the, in the case combo, what is the truncated length of the truncated guide RNA? Yeah.
Your first question was about I'm sorry, what kind of marker?
Fluorescent
I don't find any fluorescent marker.
Yeah.
When you go for a higher crop and transformation is very difficult.
Mm-hmm.
So are you thinking about to put those things so that it will be easy for the other research? Because I'm modifying the 203 and IPQ and all kind of things.
So a fluorescent marker for visualization after plant transformation, right? Is that right? Okay, so I think our plasmid doesn't already possess a fluorescent or another visual marker. However, I think it would be easily modifiable to include something like that, like a red fluorescent marker or even GFP. Certainly something that if you're working on species that are increasingly challenging to transform, that would be something to add into 203. So there's nothing precluding you from actually adapting our system. It might be something worth doing. And then I think your second question related to guide RNA length, but I might have missed that.
In the case combo, you use truncated guide RNA. What is the length?
Oh, 15 nucleotides. So when you truncate the 20 nucleotide CRISPR RNA portion of Cas9 to 15 nucleotides, it actually results in it abolishes the nuclease activity of Cas9. So then it confers DNA binding, site-specific DNA binding, but no nuclease activity. And we've harnessed that for gene activation. Yeah.
If you truncated 15, so do you think that the number of off-target will be more?
The number of off-targeting, you said?
Yeah.
Potentially, but the thing is, the off-targeting is less of a problem with genome activation than it is with, if you have a nuclease-active Cas9. Because off-targeting, it could result in maybe perhaps a little bit more because the spacer length has been shortened, so maybe you get less specific binding. But like I said, it's less of an issue or less of a concern with just an activation system because it doesn't result in any nuclease activity. Yeah.
I actually have a follow-up question for that. Have you guys tried the truncated guide RNAs with the spry version of Cas9 or Cas9NG to see if that also works similarly well for those?
Yeah, to answer it simply, yes, we have coupled the activation system with the PAM unconstrained variants, which could be useful just for targeting. The thing is with activation, it's less important where the guide RNA binds, right? Because it can bind in multiple regions in a promoter, and you can still achieve upregulation of gene expression. But if you're performing simultaneous genome editing and gene activation, maybe your editing target site possesses a non-NGG PAM, and you must edit that target site. So in that case, that's when you would opt for the PAM unconstrained variant, which can be coupled with the truncated guide for guide RNA, CRISPR activation.
So my question is very basic for beginners. So if you want, as a beginner, obviously you want to be able to one day be able to design any vector that I want for any project. What systems would you say for training purposes would be really good to give me a baseline of understanding how to do vector design, both modular and non-modular systems?
Thanks, Hannah, for that question. Sorry, to clarify, you were asking about what tools or what platforms or?
Yeah, so basically, if I want to become an expert at designing my own vectors, I know there's my lab uses Infusion too. You talked about modular cloning today too. What systems would you say would be really good to train somebody to get them really good at building vectors, both like Infusion? Which one should I try to learn before I graduate, basically?
So I would say, first of all, it'd be important to master non-modular cloning, be able to create your own level 0. For my undergrads, I've had them make a level 0, a level 1, and then transform Benthi, so Nicosia benthiama. So just as long as you can make sure that you can make your own non-modular construct and modular construct, I think that it's a great tool to learn. As for which platform, it's really up to your lab and what you already have. You don't even actually have to have a platform, but it's very recommended. Do y'all want to add anything?
Yeah, I think Katie pretty much hit the nail on the head with that. I think it's obviously it's important for the community that we have multiple different options. So we have the system that we use in our lab, and we have Gauntry, and we have MolClo and Golden Braid. I think, as Katie actually mentioned in her presentation, just choosing one and sticking with it is a great route to go. So I think any system can be a system that you can learn on. There's not necessarily one system that outperforms another in terms of just learning how to do modular cloning. But it is important because as you're constructing these vectors, you're going to find things like small errors in your plasmids that you'll have to correct. And in my experience, most of the learning opportunities come from overcoming those challenges of having to maybe just correct a vector or modify it otherwise. So I think, that's probably all I have on that.
So I have a question from the Mentimeter. This is from Karam from the University of Georgia. How do you know the efficiency of guides before transformation?
A few different methods. So the first would be sort of an in vitro assay. So you could just combine your RNP if you can source Cas9 as a protein and synthesize your guide RNA or have it synthetically prepared and then delivered to you. You can combine them and also synthesize your target site in vitro in a test tube, and then you can observe essentially degradation as if you're looking at restriction enzyme digestion, right? So that's one way to do it, but as I mentioned before, it's not, it doesn't necessarily translate to what you will observe in plants. And the reason, or at least part of the reason, is that you're not, in plants, you have an entire genome that the CRISPR machinery is dealing with, for one. And then secondly, you have the chromatin environment. And we already know that less chromatin accessible regions are more challenging to do genome editing on. And that's part of the reason that we use the protoplast assay. So if you have a protocol and your lab is capable of doing the protoplast assay, we do it in rice and tomato. That's a great way, and you can just do that in a couple weeks, really, if you can develop the plants. That's a great way to test your guide RNA efficiency, but no system is perfect for testing guide RNA efficiency, because even sometimes what we see in protoplast doesn't transfer to what we observe in stable lines. So.
Yeah, this is Sailaja from University of Georgia. So in continuation of that question, like in rice you are saying 12 guide RNAs you have used. So have you checked the efficiency before of those guides, of all those guides?
So that work was done by a former postdoc in our lab. That wasn't my work necessarily, but she did test up to 16, and I'm pretty sure that she had tested it in protoplast prior to, because that's the standard aspect of our pipeline, is to test the sites in rice protoplast prior to doing tDNA delivery and testing it in stable lines. You know. Mm-hmm. It certainly cannot hurt to test it in protoplasts. You know.
Okay, thank you.
There is one question I'm going to ask. It is beginner's question. And the question is, we know there are different kind of Cas9 or CRISPRs people can use. However, there is a limitation of genome sequence, quality genome sequence accessible to users. And so what advice you would give to a beginner, how they can go about checking or selecting the sequence they would like to modify? Because, yeah, whatever you would like to give the insight on. And then the way I go about is the plant material I'm working on, I would sequence that to ensure that sequence present. But what is your take on it?
Yeah, so just to say what I do, I actually prefer to design guide RNAs manually, and we use with rice, which has high quality reference sequences. So generally, we don't even have to necessarily amplify our target site and look at it first, because the reference genome is it's very accurate. So my approach to designing guide RNAs, if I'm interested in just doing a knockout, is towards the end of the first exon is what I would target to leverage nonsense mediated decay of the RNA. However, you can use tools. Actually, Benchling has a great tool for designing guides based on various plant reference genomes, and it uses an algorithm to sort of score the guide RNAs to tell you which one is likely to be the most efficient. If you're working on a species that doesn't have a quality reference genome, take at least the closest relative that you can, and design primers to amplify your target site and just Sanger sequence your target site prior to designing your guide RNAs, right?
Thank you.
I'm Adrienne from Simplot. My question is for Roger. So we work with gantry, and the stacking process when you're putting a lot of stacks in is quite long. Is there a way, or are you working on a way to get multiple stacks in at one time?
You mean multiple stacks into a single tDNA? Or do you mean multiple tDNAs introduced into the plant at the same time?
The stacks to get into your aggro.
So what I showed today is we now have level 2 and level 1 Golden Gate MoClo recipient donors. So you can stack multiple genes up to 6 or 7 in each of the donors at one time. And so that can be introduced in one round of gantry stacking.
Oh,
so you can easily build very large, if you have that much cargo, you can easily build very large tDNAs in as little as 1 or 2 stacks, yeah. 1 or 2 gantry assemblies.
I'm thinking more of multiple donors into?
Oh, you mean?
Yeah.
It would be hard with the existing system to design a way to make the assembly multiple component at one time, in part because of the way we toggle it between one antibiotic selection marker and another. So the importance of that switching, it forces you to recover colonies that have that specific cargo sequence added, right? So I suppose there could be a way that you could design it, but it would require redesigning not only the recipient strain, but the donors as well, where you could do multiple assemblies at once. I could see how that would make it a little quicker, but I think it's pretty easy to build big enough tDNAs now in just a matter of 4 or 5 days. You could build a tDNA as big as 5 or 12 cassettes, expression cassettes. So I don't know if it'd be worth the effort to develop a system to do that, but it's certainly possible if, the other challenge there is that the recipient agrobacterium strain, you're gonna need multiple antibiotic selection markers that are functional in that strain. So one of the challenges with RPort, that one we initially developed, is that it's sensitive to kanamycin, it's sensitive to gentamicin, but it's resistant to spectinomycin and almost every other selection marker that's commonly used in binary vectors. So it would be a challenge to do it in that particular recipient, but there might be other agrostrains that you could use spectinomycin selection or some other selection for doing something like that. It's certainly possible, but no, we haven't developed anything like that yet. So.
Thank you.
Hi, this is Daniel, University of Maryland. So for Golden Gate, one of the, I guess the biggest challenges is when you have to create parts and you have to domesticate them because of the presence of the type 2S sites. And you know, it's probably not a big deal when you're cloning a promoter. You can just change maybe one nucleotide and then have it without issue. But sometimes you get unlucky that you want to clone a gene that encodes for a protein and you have the PSA site. And then my question is, if you have faced that, and maybe it's a broader discussion around, you know, codon optimization, but do you have any guidance of, you know, how to proceed? It's not just changing one DNA base pair and maybe you change the codon usage and then your protein doesn't work the same. So can you elaborate a little bit on that?
Yeah. So thank you for that question. If it's just one BSA1 site in your coding sequence, then I would look at the overhang and see if it's something that would even interfere with your cloning, because if it's not going to interfere, you may be able to be lucky just to screen a little bit more and you could get away with it. If there's 3 or 4 BSA1 sites, I would definitely domesticate all of them. And it's dependent on if you need to do PCR or synthesize them, but as for validation of it in the plant, you could check other reference genomes and just see if you can get away with just a nucleotide change without an amino acid change. And typically with that, you may only need to validate it in your plant system.
So just maybe to emphasize again, yeah, I mean, you can definitely use any of these Golden Gate cloning methodologies for cloning into the donors for gantry, but there's no prerequisite for domesticating any parts that you assemble with gantry. There are only 2 prerequisites. One is it can't have the recombination recognition sites, which are actually large. They're around 40 to 60 base pairs. And the other is it can't confer resistance to kanamycin or gentamicin, but beyond that, any parts could be stacked with gantry if you can get it into the donor plasmids, right? The other thing I would say about domestication of the parts for MoClo or whatever Golden Gate cloning strategy, yeah, it definitely can be a pain if there are multiple sites in your sequences, the domestication. So synthesis is usually the easiest way to go if there's more than one. But if it's a single BSA1 site, amplifying it and introducing a single nucleotide change within that recognition site is pretty easy. The other thing that when you're doing it for coding sequences, yeah, which codon you're going to change to. I would look at the codon usage tables for your species and make a change where you're not changing that codon to something that's very rarely used in your species. But beyond that, I don't think there are any strong rules of how to best do that. But you could certainly, if you have the bandwidth, you could make multiple different changes to the codon and compare them and see if one of them works better than the others. But that's quite a bit of work to do just to change one nucleotide. Do you have anything to add?
Yeah, just something brief, at least because I've worked with Cas9 and had to domesticate or abolish the BSA1 sites, multiple BS1 sites even. We're not necessarily getting rid of the restriction enzyme site, but previously I'd introduced introns into Cas9 and had to create compatibility with the splice sites around the borders of the introns by introducing synonymous codons, and it still works very well. I mean, as long as you keep up with the preferred codon usage, as Roger mentioned, there are tables for that. I mean, with rice and maize are very similar in their codon usage. If you can, just use the top one or two codon preferences to domesticate your sequence. But usually, it's not really a major concern as long as you maintain the same amino acid, right? And Katie's, yeah.
So there was one time that I was personally dealing with having ESP31 sites in a prime editor that I wanted to use, and so to get around that, because I needed to go into a level 2 multi-green acceptor, I actually just kept my level 1, which then I amplified from overhang to overhang so that I could skip using ESP3I altogether. So I just amplified from overhang to overhang and put it back into a level 0 so that it wasn't really just an element, it was the full transcript in a level 0. And so that's another way that you can get around domestication if it's your secondary restriction enzyme.
I have one other thing to add. So the classic joke in the lab I was a grad student in is molecular biologists are lazy, so you try to figure out the easiest way to do things. If you know that that part you're using is going to be a one of and you don't plan to use it again, if that endogenous site has an overhang that's incompatible with all the other sites in the system you're doing the assembly, it could just become 2 parts instead of 1 part in the assembly, and you can still assemble it, right? It's just you have to check all that before you do it, but you don't always have to domesticate it. You still potentially could get the assembly, but you got to make sure that the endogenous overhang that's present for that restriction enzyme in that part doesn't want to ligate with any of the other overhangs you're using in that particular assembly. Yeah, that's the lazy way to do it if you don't want to domesticate it. Yeah.
Yes, I'm Chuan from the KWS. I'm almost working in the transformation regeneration, so I have the simple question for the editing plants efficiency purpose. Do you all have any suggestion for the user vector? How the one vector including the nucleus and the guanine is work better or separate the nucleus and the guanine with different vector is better? What kind of strategy you all can give us for this purpose?
I might have misunderstood your question. So it's relating to selection of the nuclease and the guide RNA vectors?
I'm focus the editing efficiency purpose. What kind of vector strategy is better? So use one vector including all the molecule components together or separated, like the nucleus CPF1 one vector or other guanine several vector co-transfers better, or what kind of strategy you can give us?
I think the question is relating to when you deliver the vectors, is it better to include them all in one tDNA, for example? Yes. I would argue yes, and the reason is because you can get several different tDNA integrations in various loci, and that could cause kind of not a very well cooperated expression between, for example, your Cas nuclease and your guide RNA. So it's better to have them integrated into the same locus together. Moreover, it reduces the complexity of having to introduce several different vectors at the same time, right? It's much easier just to introduce it all in one. And these CRISPR vectors aren't too large either to do that with, right? Compared to like the gene stacking stuff. So I would, if you can, just all in one, yeah.
You mean all the one? How many guide RNA can put in one?
You can put many guide RNAs into one vector. We've demonstrated up to 16, for example, but you can go beyond that, certainly. Yeah.
Okay. Thank you.
Hi there. Oh, sorry.
Go ahead, go ahead.
I'm Freddie from Purdue University, working with Dr. Kranti Varala. Each of you, I think, touched on it briefly in your own talks. You talked about the way you save files, the way that you maintain this flow of information. As we've seen, I'm blessed enough to be a fifth year, and I've seen many master's students and postdocs come and go. We often have a loss of continuity in where things are stored or kept, and it's said many times to be a problem. I have no doubt that you guys have encountered this too in your own ways. What is some advice that you have to kind of maintain that lab-level knowledge of all of these great resources that are made?
So one thing I learned when I was a grad student a long time ago now is that, yes, that continuity is really hard to do if you have multiple members of the group and they all have their own stocks and their own records, their own ways of keeping it. So I learned back then that one common -80 glycerol stock file, a spreadsheet that contains everybody's validated constructs in an organized fashion and organized, as Katie showed, in specific stock boxes that are in the -80 in a particular numbered order. That's absolutely important. And one thing I would add is that spreadsheet, if it's Excel or whatever spreadsheet system you want to use, or if it's in the cloud even, keeping all that organized and making sure that everyone that's a user of it knows how it works, and they only put validated, confirmed clones into it. And with that, there's a very specific description of what that vector is. And then the way we do it now with like SnapGene or whatever, then we keep a separate folder that has the SnapGene file for that particular construct with that stock number associated with the SnapGene file. So it's definitively associated with that specific sequence. And now with whole plasmid sequencing, it should be perfect for the entire plasmid. So that organization is really important. When we adopted Golden Gate cloning, so I had that type of system for all the vectors and strains we had in our laboratory. And when we adopted MoClo, Golden Gate cloning, I didn't want to try to make stocks to add all of that to our existing lab file. So we created a separate file that's a stock collection now for all the level 0 and level 1 parts. That is the stuff that you'll just use to build something new using the MoClo approach. But anything we put into plants, that stock will go into the main lab file. So anything we would transform into plants would be in that one, because those aren't modifiable at that point. They're at a fixed endpoint. So that's how we organize and keep things separate, and making sure all of that stuff is connected and that everyone in the lab knows how it works, knows what the expectations are, and that all those things are connected together with sequence, stock, everything. Even some people put the lab notebook page, whether it's digital or physical, in the stock file too, so you can even go back to their lab notebook and look it up and see exactly what they were doing and why they were doing it. It's easiest to do that when the lab is just new and getting set up. It's really hard to establish that system in an existing lab that already has lots of stocks. So I don't have any advice for how you would do that for a lab that doesn't have a system like that, but anybody that starts their own lab, I would strongly encourage you to do that type of organization. Katie?
I will add to that really quickly that if you're looking for an example of an Excel sheet like that, I did put one on my blog with fake data, but it says, just saying, but it does say like you should have the box location, which box is it, which column, row is it, so that it basically has map coordinates to each plasmid. Then you can have a description of that plasmid, the name, the person who did it, or the date that that person did it, and then, like Roger said, everything in your freezer stock should also have a corresponding plasmid map or sequence that you can have continuity with. Anything to add?
That's good.
Good morning. My name is Sandra Romero. I am from Connecticut and Verinomics. So my question is regarding the backbone at the end. When you are planning the construct, do you have any recommendation about the orientation of the cassettes that you use for the Cas protein, the fluorescent marker, the selectable marker?
Sure. So left border is where you want your selection to be, your resistance. And some people even have multiple borders for that. Like they'll just have their last level 1 or whatever be just multiple levels borders so that it can ensure that only one tDNA is inserted. So typically we'll keep our resistance as leftmost as possible and our visual selection one way.
So I think another part of your question was maybe the orientation of the transcriptional units, the expression cassettes. Yeah, that's something I intended to talk about, but I think I forgot in my presentation. So if you look at most of the constructs, the 10 stack or the 11 stack that I showed, the orientations of almost all the cassettes was direct. So start to the end of the tip of the arrow in direct orientation. Whether it's this way or that way in the tDNA, I don't think that's so important, but we typically try to avoid where you put convergent expression cassettes. That is, 2 expression cassettes are directed toward each other. The rationale for why we do that is sometimes it's well known that the transcription terminators fail to terminate transcription, and if you have directly cassettes expressed toward each other, actually, especially if they're really strong constitutive promoters, they could not only transcribe the gene of interest, but continue past that transcription terminator and start making antisense transcript of the gene that's downstream. And you definitely don't want that to happen. That's well known to activate silencing. And so what we almost always do now is put everything in direct orientation. Everything's oriented the same way. We arbitrarily picked the orientation such that when we look at the maps, it's left to right, because that's how I usually think about it, promoter, gene, terminator. That's just the convention that we do. But there's no rule saying you couldn't do divergently oriented cassettes. One directed this way, the other directed that way. That's fine too, it's just then you got to follow that orientation after that. After those 2 are stacked together, if you stack another one, it has to follow the right orientation so you don't get a convergent cassette. So that's what I would recommend. And I think it's just simpler to just pick the orientation you prefer and just build everything in that orientation, because then you don't have to think about it again. It's always going to be in that orientation. Then I think that's just simplistically way to do it.
I'm good. I think we have a question online.
Before we take another in-person, let's give the Mentimeter questions a chance. Amy, do you have any Mentimeter questions?
I do, yeah. So I have 2 questions. I'll just ask the first one. It says, this is for Katie. What is the successful rate for 16 guides assembly compared to 2 guides assembly? Yeah, I think that might be for Josh.
Assembling the guides, actually, it's a slightly different system that we use. We have methods, protocols that are published for doing this. It's not less efficient because we use a combination of both Golden Gate. We actually do Golden Gate of like 4 guide RNAs, 4 guide RNAs, 4 guide RNAs, and 4 guide RNAs, and then we combine with restriction enzyme digestion ligation. So you can't really compare it very well to sort of the original method, but it's efficient and it's not challenging necessarily to assemble constructs with that many guide RNAs. Yeah.
I'll add. Yeah. So you can definitely do Golden Gate-based assembly of many parts. I've noticed personally in our group that when the parts get larger and you have many parts that are multiple kb in size, the efficiency of the cloning definitely does reduce, right? So assembling small parts like stuffer elements or small parts, yeah, you can do 52, but you're not going to, like NEB, New England Biolabs has done, you're not going to do 52 parts of multiple kb units, right? So definitely that does affect the efficiency of the ligation reactions of all those larger parts. So I was just trying to build something that had 5 parts, 5 cargo parts and an end linker to it. And I struggled because we use homemade component cells. We don't buy them commercially, we just make them ourselves, and they're only 10 to the 6th, maybe 10 to the 7th component. If you had 10 to the 9th, I'm betting it would work fine, but we struggled at that normal competency. But I broke it into 2 parts, put it in a B donor and a P donor, all the components, and that assembly worked a lot better when I had fewer parts in each assembly. So there is some efficiency trade-off for large cargo parts with Golden Gate cloning, at least in my experience. Got another one? Oh.
Amy, was there a second part to that question, I think?
No, just I have a separate question on here entirely. So my question is, when considering selection of promoters for guide RNA expression cassettes, does the promoter only affect the expression of the guide RNA? Is it possible for the promoter to affect the guide RNA structure?
So generally, there are 2 promoter strategies. The first one we refer to as the polycistronic RNA promoter strategy, or single transcript unit, using a constitutive polymerase II promoter. So that does affect the structure of the guide RNA itself, but that's intended. So it forms tRNAs because each guide RNA is expressed in one large mRNA, and then between each of those guide RNAs, there's a separate tRNA, which is then processed by endogenous machinery in the cell to produce individual unique transcripts for each guide RNA. So, and afterwards, the guide RNA takes on its native or intentional secondary structure with this, the stem loops that you're interested in having. The second way would be to use the POL3 or polymerase III strategy, using the small promoters U3 and U6, and those produce guide RNAs also with the desired secondary structure harboring the stem loops. So I think that answered the question. Anyone else have? Okay.
There was a question about laboratory management of plasmids, and so I do want to plug in there is a session which is led by Wayne Parrott, stewardship and compliance of materials. And then this is whatever we do with the plasmid, it is very important to do like track and trace and quality check. And then, because that is the foundation for any transgenic material we produce, so I will highly recommend you to check on that session. And there is a question here.
Hi, this is Tim Olmosoff, Sequesta. Great session, by the way, great panel. I'd like to ask the same question I was asking 2 years ago, I believe, at this meeting, which is like if somebody is doing gene editing, CRISPR machinery has 2 components, Cas9 protein and guide RNAs, RNA, and they both have to be expressed, right? And if you use an RNP, they're equimolar, and there is no question sort of about which one is more efficient. But if you have to overexpress it, which one is rate limiting? I think for, if you are a researcher, pretty much you're doing transformation, you get maybe 10, 15, transformants, and they usually vastly different in the ability to express or, produce your gene edits, right? And there could be 2 or 3 that are, doing it very, very high frequency. And typically, most people don't understand what's going on. So I was always asking myself, is it expression of the nuclease that's rate limiting, or this generally expression of the guide RNA rate limiting? And 2 years ago, I could not get a satisfactory answer to that. Yeah. And maybe you can also think about or maybe speculate about what's going on when you get these different transformants, why they differ so drastically in their editing efficiency.
Either of you? I'll take it. I'm going to give you a very hand-wavy answer that probably won't be sufficient, but I would say I don't think we know for certain which expression component is the rate-limiting step, for example. One hypothesis would be that because Cas9 is both has to be transcribed but then also translated, that could be more rate-limiting compared to just the guide RNA, which simply needs to be transcribed, right? But one way to sort of overcome this is to use coordinated expression with the same promoters. So the same pull-2 promoter and this driving Cas9 and the same pull-2 promoter driving the guide RNAs, and at least then you can get perhaps a more higher level coordinated expression of both reagents at the same time. However, I'm not sure exactly, I don't think we know as a community which exactly is the rate limiting step. I think when you get to lower editing efficiencies, I don't think it's due to the molar ratio between the nuclease and the guide RNA. I think there are other factors that are involved, right? Temperature, chromatin accessibility, other things that are more challenging to explore. But as you mentioned with RNP, it's just a simple 1-to-1 ratio. With expressing these systems in vivo is more complicated. It's hard to point what really is the rate limiting. Yep.
I'll add one bit of an argument to this. I think by multiplexing the guides and being able to get multiple activities at all those target sites, it's arguing that the guide is not usually limiting the total activity when you're giving it just 1 or 2 guides. It must be the activity of that editing protein, would be my argument. The other argument that would support the idea that it's usually the Cas editor that's the limit is that when you compare Cas9 to all these other versions, Cas9 is just super active comparatively, right? So that has to be a protein attribute of that particular editor. And so I would say in most experiments, the limit is either the inherent activity of that guide or how much of that editing protein is being produced in that particular cell. That would be my argument. Yeah.
Hi, it was great to hear all your talks. Letitia DeRose, I'm from Agriculture and Agri-Food Canada. I just had a question just in terms of stacking when you're putting multiple proteins together, just input for this in terms of separation of those proteins. There are a couple different mechanisms like the ribozymes or the P2A sites, which also requires the addition of another protein. If you guys had any comments on uses and efficiencies of cleaving those.
Did you? Sorry, I think you mentioned P2A. Yeah. So those are the so-called ribosome skipping. We used to call them self-cleaving. It turns out they don't really self-cleave, that it's actually a lack of the formation of a peptide bond between the two elements that are separated by either P2A or T2A. In my experience in rice, I've used T2A extensively to express several Cas genes at once in what we call cascade, involved in a type 1 system. So we express Cas3, Cas6, Cas8, Cas7, and Cas5, and we separate those using T2A, and that works very well in rice. We also, many of us have expression using Ruby, which if I recall is 3 genes, and we also use the ribosome skipping strategy to separately process those out of a single transcript. And that works very well, at least in my experience in monocots. I can't speak for many other species, but yeah. Does that answer the question, or did you have? Mm-hmm.
Yeah, so I've also seen like the hammerhead ribozymes used. Yeah. So more of like a comparison if you guys have looked at different systems for that cleavage and how they may have compared.
Yeah, so usually the ribosome skipping strategies are for separating peptides, or coding sequences rather, yeah. Whereas the hammerhead ribozyme, that works very well. So we use that, for example, with Cas12a. At the 5' end of the spacer, we include hammerhead, which cleaves more at the 3' position. And then we use HDV, the hepatitis ribozyme at the 3' end, which cleaves at the 5' position, so that it cleaves out the guide RNA in a very clean and predictable manner, which is important for Cas12a because it's more sensitive to, the mismatches or such at the guide RNA positions. Separately from the hammerhead HDV, we also use the tRNA strategy. We use that mostly with Cas9. So we separate the guide RNAs using the tRNAs, which are processed endogenously, separate from ribozymes, which self-process, right? They self-cleave. The tRNAs are processed by, I think, RNase P or an RNase D, perhaps, in eukaryotes. That also works very well, and it turns out that the tRNAs actually have some level of promoter activity, which could be viewed as kind of an advantage of using the tRNA strategy. It just boosts the expression a little bit of your guides, but maybe, I don't know.
We have just a few minutes left. I suggest we take one more question from Mentimeter before we wrap up. Do we have any other ones? No more. Okay, anybody? Oh, Venus.
I am gonna make a comment, and then Amy can speak more about it. Is anybody who was not able to ask question? Amy, can you please tell attendees about the discussion forum so that they can follow up, so that we can have this conversation going?
Thanks. Yeah, so I'm just going to repeat a little bit of what Joy said at the beginning. So we do have a discussion forum on our website. This was, the members asked for it, and we were able to deliver with the help of some amazing volunteers. So if you go to plantgene.sivb.org, and you'll need to log in. You'll see the discussion forum, and there's a general discussion forum for just all kinds of questions. This is it's an email notification system, so it gets sent out to everyone who's registered. Right now we have over 1,200 members, so there's definitely someone out there who can help you find a solution to your problem, or someone who's maybe just happy to discuss and bounce some ideas with you. Yeah, so I encourage you to use that. Thanks.
So we are going to, well, Amy doesn't know this yet, but I'm going to ask her, send out a kind of questionnaire at some point after this meeting to the PlantGENE members to give us feedback on what you thought of this workshop. All kind of comments, what you can see as improvements that really helps us, what you liked best, and any suggestions for next year. It doesn't have to necessarily be cloning. Our very first one we did in 2023. We did it on the kind of do's and don'ts of plant tissue culture and transformation, and it was also as a panel discussion. And that video is available on our website. And then last year was the basics of vector design, and then we had the modular cloning this year. So it doesn't necessarily have to be about cloning, but we did that because you guys asked us to. So please, when Amy sends that email, to complete the survey, because it really does help us. So I think we had some really fantastic speakers, and I think they did an amazing job with all those questions. So thank you all for coming and for your questions and for staying around and making this a really engaging workshop. And let's give a big round of applause for our speakers.
Thanks. And then just one more thing before you go. If you sat in the back and you didn't get a button, they are on the tables up front. Not only are they an excellent fashion accessory, but they also help spark conversation just throughout the conference. It's easy to identify the plant people and, yeah, strike up a convo. So please come get them.